Well, good morning. We invite you to take your seat. Let's start. Well, welcome to the nutrition section. My name is Luis Corona. I am the moderator. We have our first keynote conference. Our first speaker is Dr. Ryan Davis. Dr. Ryan Davis, based and raised in Pembrokeshire, West Wales, United Kingdom. Graduated with a bachelor's degree in veterinary medicine and surgery in 2002. He spent over 20 years in clinical farm animal practice before joining to SCCL as a veterinary technical consultant in October 2023. His passion is for preventive hair health medicine and reducing antibiotic usage on farms. Welcome, Dr. Davis, with the conference, World Colostrum for Cow Welfare and Long-Term Productivity. Thank you. Go ahead.
Thank you. Good morning, everybody. Okay. Today I'm going to show you the difference between this calf and this one. These are two calves born on my study farm this year. They're both born within a day of each other. They both are within 100 grams of body weight, and they have the same sire. So the only difference between these two animals is 50% of their genetic material and colostrum. So today we're going to talk about whole colostrum for calf welfare and long-term productivity. The talk is divided into three sections. We're going to talk about antibodies and immunity first. Then we're going to move on to colostrum as a source of energy. And finally, we're going to talk about lactocrine signaling for growth and development. So there's a lot to get through, so bear with me, please. So what we all know, colostrum was first documented to be essential for the newborn calf in 1922. Calves are born A-gamma globulin-anemic. So without colostrum, this study showed that they were susceptible to E. coli septicemia, and the mortality rate was very high. So we know colostrum's a necessity. It is not a choice. For numerical standards, several studies over the years have shown that a statistical difference of immunity over and below 10 grams per liter of serum IgG. So there we formulated the phrase failure of passive transfer, being below 10 grams per liter, successful being over. But as you can see from this data that was taken from NAMS in 1993, that we still have a 3% mortality even if we're over 10 grams per liter. And this doesn't take into account morbidity, doesn't take into account antibiotic usage or welfare or anything like that. Figures from more recent from the US show that failure of passive transfer still runs at 19% with dairy and 33% for beef. So this bar is pretty low, and we're still seeing a high rate of failure of passive transfer. So what's going on? How much IgG, how much antibody do we need to achieve this 10 grams per liter? This is a study done in 1998, and it showed that in the first 24 hours, to achieve 10 grams per liter in the serum, we need to administer 100 grams of IgG to a calf. But we know that maternal colostrum can be as much as 30 bricks, which would be 100 grams per liter, and that would tell you that this calf would only need 1 liter to achieve this. So why is there still such a high rate of failure of passive transfer? So in order to answer this question, we need to divide it into two sections. Those are the dam. So we're talking here about colostrum quality, which we divide into antibody mass and contamination. In terms of antibody mass, we term good quality colostrum as anything over 22% bricks, 50 gram per liter. In terms of contamination, we want the total bacterial count to be below 100,000 colony-forming units per mil, or 10,000 coliforms. And then we must look at the factors involving the calf, and we term this apparent efficiency of absorption. By that, we're talking about the percentage of antibodies we administer to the calf that make it stream. There's many factors that influence this: timing, contamination again, method of feeding, and stress. And I'm including handling stress within that. So we'll have a look at these now quickly in a bit more detail. In terms of colostrum quality, transport of IgG into the colostrum is done by a receptor-mediated transport mechanism, which is genetic. So once that transport receptor is saturated, then no more IgG can go into colostrum. We know that there's an environmental impact. Heat stress, that's cold or excessive heat, can reduce the quality of colostrum in the last 2 weeks. And the big one in my experience on farms is the delay to harvest in the colostrum. It can go down in quality by as much as 3.7 bricks an hour when it's not harvested. Now, this is due to a variety of factors: leakage, possible resorption of antibodies back into the maternal circulation, and several other factors. Then seasonality. We know that colostrum quality is linked to day length, so when the day length shortens, quality can go down. There's other factors that are influential, such as parity of the dam. You can see that heifers have lower quality, and a few other things. So just to emphasize the point, we look at this study in an Italian dairy that was done in 2018. On the left-hand side, we have a graph of colostrum quality. The white bars being good quality colostrum, over 50 grams per litre. On the right, we've got the corresponding dairies and the transfer of passive immunity rates in the calves. The black bars being a failure, below 10 grams per litre, and the grey bars being successful passive transfer, and the white bars being very good passive transfer, over 16 gram per litre. So if we look at these two dairies here, dairies 11 and 12, you can see that 100% of their colostrum is of good quality. Follow them across to the right, and you can see that they've got a 50% failure of passive transfer rate. So even though they're producing good quality colostrum, there's something else involved here that's causing a persistence of the failure of passive transfer. So what would this be? Then we've got to look at factors with the calf. Apparent efficiency of absorption, on average, would be about 30 to 40%. 50% would be absolutely excellent. What factors influence this? The big one is timing. You can see that in the first 2 hours, absorption of antibodies is at its maximum, but it's still below 50%. It tails off quickly after 6 to 8 hours, and then by 24 hours, we've got gut closure, where the gut is no longer permeable to absorbing macromolecules. Several factors will affect this curve. For example, twin calves have a lower absorption. Dystocia is the big one. The absorption rate can drop as much as 30% if we have dystocia, and that curve would look like that. So for those animals, we'd have to administer more IgG. Just a word about method of feeding. Using an esophageal feeder, obviously we bypass the esophageal groove reflex. And if you're feeding low volumes, that can lower absorption, because a lot of the colostrum will end up in the reticular rumen. But if we feed over 3 liters, then it's not a problem. So bacteria is the big one. It can affect antibody mass, and it can affect absorption of the calf. If colostrum is heavily contaminated with bacteria, the antibodies will bind the immunoglobulin molecules in the lumen of the intestine and reduce the mass that's available for absorption. They will also reduce the absorption capability of the calf by damaging the enterocytes and reducing the transport of macromolecules across. They also block the binding sites. And of course, then this will accelerate gut closure. So here's a couple of pictures from my study farm. When we started, this is the colostrum harvesting equipment. So you can see the level of contamination there, even if you have top quality colostrum. Milking it into these and then administering it to the calf, we're not going to get good transfer of passive immunity. So as I say, standards for colostrum quality, 100,000 colony-forming units per mil total bacteria and 10,000 coliforms. Coliforms are worse. Also remember that there are certain pathogens that can be transmitted through colostrum: E. coli, Salmonella, Mycoplasma, Johne's disease, and TB, which is a big thing back home where I am. A study was done in 2012 where they fed 2 groups of calves the same pooled colostrum, but one group it was heat treated. And what they found was that the disease incidence in the heat treated group was less. And rather than attributing this to reduced infection straight from the bacteria, they attributed it to a higher rate of passive transfer. These animals had an average of 18 grams per litre IgG as opposed to 15. So bacteria is critical. So some work was done by a panel of calf experts back in 2019 just to ascertain some new standards for transfer of passive immunity. From their results, where they looked at the most recent data, if you look at the yellow line, which is concerned with mortality, you can see that drop below and above 10 grams per litre serum IgG. So that agrees with the traditional standard. And you can see as you go further up, there isn't really any statistically significant change. The most significant finding was the grey line, which is the actual morbidity. And you can see as you go along the x-axis, increasing the serum IgG, you can see that there is a corresponding decrease in the morbidity. So the more IgG we're able to get into the serum of the calf, the more protection they've got against diseases. That is going to translate then to improved animal welfare and reduced antibiotic usage. So they came up with this table based on the recommendations, where there's 4 categories. So the poor category would be the traditional failure, below 10 gram per litre. And then we have fair, good, and excellent, which would be above 25 grams per litre. So they recommended that we should target 40% of our animals to be in the excellent category with only a 10% failure of passive transfer. If you want to know what that looks like in real life, going back to our girls at the beginning, calf on the left was excellent passive transfer, and the one on the right was a failure. As you can see the difference. So how much IgG do we need to feed in the first 24 hours to get excellent passive transfer, 25 grams per litre? Going back to our study that was done in 1998, we can see 100 grams gives you your 10 gram per litre, as we've said. If we want, with all the factors we've discussed, if we want to achieve 25 grams per litre on average, we need to feed more than 300 grams of IgG in that first 24-hour period. So just briefly then, just going to go over the consequences of poor colostrum management practices. I'm sure you're all aware, obviously we're going to have a higher incidence of disease. And as veterinary practitioners, we know that if we have a high incidence of disease, we're going to have to deal with some mortality. And we're going to have to increase our antibiotic usage. And then our animal welfare is going to suffer. And I think antibiotic usage and animal welfare are major considerations that we have to look at moving forward. Obviously then, we're going to have reduced daily weight gain. Having diseases will, by definition, reduce our daily weight gain. And when we look at the adult animal, poor colostrum management practices have been shown to produce a delay in calving of heifers, which is obviously costly to the farmer. Then lactation performance is suboptimal, and reproductive performance. They take longer to go back in calf when they're into the herd, and they're more likely to be culled in the first lactation. So when we look at all these things that we've got on this slide here, is that purely due to antibodies? Is that purely due to the antibodies we're administering in the first 24 hours? No, it's not. There's other things that are involved. So we're going to move on to the second section now, which is the section on colostrum as a source of energy. So the neonatal calf, when it's born, can survive for 15 hours with endogenous lipid, or 3 hours with endogenous glycogen reserves. After that, they will utilize amino acids, glutamine and alanine, to synthesize glucose, and then you're into protein catabolism then, so we're losing weight. So giving that source of energy to calves in that first 24 hours is vital. So colostral fat, what does it do? Well, it's a substrate for thermogenesis, so it's very, very important. And also it's a substrate for gluconeogenesis, so it provides that energy source. It's also been shown that fatty acid oxidation can improve gastrointestinal health. We'll touch on that a little bit more in a second. So the milk fat globule, or the colostral fat globule, what is the structure of it? We have a central triglyceride core where 98% of the fatty acids are located. Then we have a phospholipid monolayer around the outside, and then a phospholipid bilayer, which is formed on secretion from the secretory cell. We call this the milk fat globule membrane. Within it, we have phospholipids, we have other polar lipids. We also have transmembrane proteins located throughout, and cholesterol. So the milk fat globule, the colostral fat globule, is much larger than the milk fat. There is more triglyceride there to act as a source of energy. And what other ways does the colostral fat globule differ from the milk fat globule? So if we take a look at this heat map, this is breaking down the fatty acids by the chain length. So at the top, we're looking at the saturated fatty acids, and at the bottom, we're looking at the polyunsaturated fatty acids. So the darker red color indicates an increase in milk compared to colostrum. So as we go across, we're going from milking 2 across to milking 12. You can see that the short chain saturated fatty acids are increasing, especially butyric acid and caproic acid, which is the C4 and C6. Interesting thing to note here is these saturated fatty acids, these short chain saturated fatty acids, are required for epidermal differentiation. So if they were in high amounts in colostrum, we're going to accelerate that gut closure. And as we go up in chain length, you can see that the longer chain saturated fatty acids are in a higher amount in colostrum, and they decrease into milk. Of course, longer chain fatty acids have got more energy locked up within them, so they are a better source of substrate for thermogenesis and gluconeogenesis. Monounsaturates are the same in colostrum and in milk, but when we look at the polyunsaturated fatty acids, these are mainly located in the milk fat globule membrane. And they are in higher amounts in colostrum, and then they decrease in milk. Most notably omega-3 and omega-6. Omega-3 is responsible for the fluidity and the plasticity of the plasma cell membrane. It's required for brain and retinal development and the development of the immune system. So we can see there that this study was done that showed that there's only a 50% overlap between the proteins located in the colostrum fat globule membrane and the mature milk globule membrane. Also, those proteins that are interspersed in the colostrum globule membrane, some of them are lactoferrin, immunoglobulins, and other antibacterial components. So that's how breaking down these fat globules will improve gastrointestinal health. So just want to briefly touch on a study just to emphasize the effect of the fat. This study was done in 2020, where they looked at the effect of colostral fat on thermogenesis, vital signs, growth, and health. 2 groups of calves. The only difference was one group was fed a full fat colostrum replacer, 22% fat in the dried form, and the other group had a defatted one. The other components were the same. And what they did was they looked at the vital signs, heart rate, respiratory rate, rectal temperature, activity of the animals. They also tested the blood to reference the transfer of passive immunity, and then they recorded disease incidence and body weight. Most significant was the difference in growth between the 2 groups of animals. The control group, or the ones that received the full fat colostrum replacer, by 127 days of age, they were on average 10 kilograms heavier. So this shows the significance of the fat, and that was statistically significant. So when they looked at health, although because of the small sample size, it wasn't statistically significant, you can see there was a much higher incidence of respiratory illness in the defatted group than in the control group that received the full fat colostrum. Now, most interesting here was the defatted group had, the control group actually had a lower level of transfer of IgG, of transfer of passive immunity. So that was significant. So the conclusions from that study, full fat colostrum promotes thermogenesis and body weight gain, and the incidence of respiratory disease was reduced. Okay, so we've touched on antibodies. Now, antibodies are significant after day one. They're not just important for the first 24 hours. We know that antibodies provide local protection. I like to think of it in terms of the castle and the moat theory. So if we're going to defend against pathogens, the moat is your local protection around the castle. If we can stop them there before they get to the gates, which would be your systemic immunity, then all the better. Colostrum also contains trypsin inhibitors, which reduce proteolytic degradation of immunoglobulins and peptides. So antibodies are significant after day one. We've touched on colostral fat for thermogenesis, gluconeogenesis. The other thing I haven't mentioned is vitamins A, D, and E are fat-soluble vitamins. So they are absorbed in very minimal amounts across the placenta. So when the calf is born, it's completely reliant on the colostrum to obtain these vitamins. What is the significance of that? We've all seen in practice hypervitaminosis A. Hypervitaminosis A. I don't know what that was about. Which causes blindness in calves. It's easily treatable, but the incidence is higher than we expect. And also vitamin E deficiency. It causes a nutritional myopathy, white muscle disease, which is not very responsive to treatment. So it shows how, these vitamins are fat soluble, so without the fat, the calf will not get them. So what else is in colostrum? So now we're just going to move on to the section on lactocrine signaling for growth and development. So here's a table that I just took from a study carried out in 2000, where they're just looking at different constituents as you go along in number of milkings from colostrum to mature milk. So immunoglobulin G, we know it's very, very high in the first milking, and then it drops off to negligible amounts in mature milk. Lactoferrin is an iron-binding glycoprotein. That's found in colostrum in about 75 times the amount that it's found in milk. Insulin, growth hormone, and insulin-like growth factor. These are all found in much higher proportions in colostrum than in mature milk, as you can see. There's other growth factors like epidermal growth factor, transforming growth factor. The transforming growth factor actually is involved with the synthesis and repair of DNA and RNA, fibroblast growth factor, and platelet-derived growth factor. So how do all these have an influence on the growth and development of the calf? They have an influence in endocrine ways and also in local ways. So if you all remember the somatotropic axis from college, regarding growth hormone, basically growth hormone releasing factor is released from the hypothalamus in response to hormones postprandially and also during the night. This goes to the anterior pituitary where growth hormone is released. Growth hormone acts directly on the somatic tissues and the organs to promote growth and development, and also on the liver to release IGF-1. So guess what's in colostrum? Growth hormone releasing factor, growth hormone, and IGF-1. So the growth hormone releasing factor is going to stimulate endogenous production of growth hormone, which will stimulate IGF-1 release from the liver and also have a direct effect on somatic tissues and organs. The growth hormone that's in colostrum, some of it gets absorbed and acts to stimulate the endogenous endocrine system of the calf. IGF-1 doesn't get absorbed into the calf circulation. So what is its purpose? Growth hormone and IGF-1 also act locally. So if we look at the apical surface of the small intestine, there are receptors, specific receptors for growth hormone, cholesterol, insulin, and IGF-1. So these substances act locally to promote growth and development. They cause proliferation, differentiation, growth of enterocytes, and this will lead to a larger and more efficient absorption surface. So you can see that the IGF-1 is not absorbed into the circulation, but it's acting locally. And you can see that these hormones have an effect on the endocrine system and locally on the gut. So here's one study to emphasize that. 2 groups of calves. They were fed for 5 days and then culled, and histology was done on the gastrointestinal tract. So you can see that one group received colostrum within the milk powder, and the other group did not receive it. And you can see that the villi circumference and the villi height is much more advanced with the colostrum supplementation. And this was only at 5 days old, remember. The interesting thing I find about this study is the group without the colostrum extract, they administered supraphysiological levels of human IGF-1 orally in the milk powder. So 10 to 12 times IGF-1 that would have been in naturally in colostrum, they administered in with the milk powder. And still, you can see these animals did not achieve the same development of the GI tract. So why is this? It's several factors. The IGF-1 in colostrum is bound to binding protein, so it reduces its bioavailability to protease enzymes. The colostrum obviously has trypsin inhibitor in it, which will reduce proteolytic degradation. But also there's other factors within colostrum that work in synergy with the IGF-1. You can't just administer one component of colostrum and expect the effect. For example, vitamin A. Vitamin A reduces programmed cell death at the tips of the villi, so it acts in unison with IGF-1 to reduce the degradation of the villi in harmony with the proliferation. And also lactoferrin has been shown in studies to also work in harmony with IGF-1 to enhance growth and development. And of course, in colostrum, we've got the growth hormone as well, and we've got the insulin also having an effect. So taking one hormone on its own and administering it doesn't necessarily have the same effect as the full package. This study, again, this is a well-known study carried out in 2020, where these calves were actually killed at 3 days old and histopath done on them. One group just had mature milk, another group had 50-50 colostrum and milk, and the third group had straight colostrum. And you can see the histology there of the ileum, the development of the villi in the groups that have had colostrum and 50% colostrum is far exceeds those that just had milk. There was no statistical difference between the colostrum group and the 50-50 group. But you can see that's just at 3 days, you know. So when I was researching for this project, I came across several studies where they were talking about the Olympic athletes using bovine colostrum powder as a training aid. Reason for that is IGF-1 is one of the most potent anabolic substances known to man, so it increases lean muscle mass and reduces fat. They also found that increased their endurance and reduced the soreness after exercise. But the World Anti-Doping Agency stepped in because obviously IGF-1 and growth hormone are banned in competitive sports. They stepped in and advised against the use of it. It's not banned, but they advised against the use of it because these substances were showing up in the blood, and obviously then they would fail a doping test. But they were only taking 40 grams of colostrum powder a day. That's a very, very small amount. So can you imagine the effect of the volumes we're advocating for calves? Can you imagine the effect for growth and development for them? I just found that interesting. So this is a table I found, basically breaking down the components of colostrum by molecular weight. We've covered immunoglobulins. We've also covered growth factors. You can see the many different types of growth factors that are there within colostrum. Next, I want to talk about oligosaccharides quickly. Oligosaccharides are polysaccharides with a sialic acid attachment. So sialic acid has been shown to bind pathogens and toxins. Where are they located? If we look again at the apical surface of the small intestine, they are located in the mucus layer along the lining of the intestine. They're also located in their free form within the lumen of the gastrointestinal tract. What do they do? I've got an animation here just to try to show you what their effect. It's threefold. So they act as a carbon substrate for commensal bacteria. The commensal bacteria then multiply in numbers. And what that means is then they occupy the binding sites and reduce the availability for pathogens. Second of all, they block the binding sites on the epithelium directly. So again, they reduce the availability for pathogens. But also, they have an affinity for pathogens with a binding site whereby they can bind pathogens in the free form in the lumen of the gut and then excreted from the system. So when we talk about, I think it's interesting to note that when we talk about transfer of passive immunity, anything that maintains the health of the enterocytes in that first 24 hours and reduces bacterial damage increases your transfer of passive immunity. So it gets quite complicated because when you say, how do I improve the transfer of passive immunity, there's a lot of components within colostrum that assist in that because they maintain the health of the epithelium. So the types of oligosaccharides, there's over 40 different oligosaccharides in colostrum. The most common is 3-sialolactose. We also have 6-sialolactose, 6-sialolactosamine, and di-sialolactose. Now I put this slide up just to show that it's been proven that 6 linked sialic acid oligosaccharides have a greater affinity for the E. coli K99 adhesin binding site. And you can see there the 6-sialolactose, increases with the second milking and then maintains its level quite far into the milking process. Now this may be a natural phenomenon that when the antibodies are decreasing, levels of this oligosaccharide are increasing in order to defend against E. coli K99. So again, another aspect that controls pathogen multiplication. This is a study done that just showed the effect of delayed colostrum feeding. Colostrum fed immediately in the first bar and after 6 hours the second and after 12 hours in the third. And you can see that the proportion of bifidobacterium especially and lactobacillus is much greater when colostrum is administered immediately. This is because the oligosaccharides provide a substrate for the commensal bacterial growth. And of course, if the commensal bacteria are there, they occupy the binding sites and they reduce the availability to pathogens. So why are we concerned about the commensal gut microbiome? Well, first of all, we know it's essential for digestion. We're dealing with ruminants here. The microbes produce enzymes which allow microbial fermentation. And the byproducts of that are there for fatty acid, protein, and carbohydrate synthesis for the ruminant. So they are required for the nutrition of the animal, but also for immunity. We know that the commensal bacteria, like I say, bind the binding site. They occupy the binding sites and reduce the availability to pathogens, but they also stimulate the immunity of the animal. They stimulate IgA secretion, which obviously acts locally as a defense mechanism. They increase the production of glycoprotein in the mucus, and they increase the production of lysozyme, which is antibacterial, and that's released from the Paneth cells in the small intestine. So the good bacteria are helping defend against the bad bacteria, for want of a better description. Also interesting to note that one of the first colonizers of the intestinal tract, Bifidobacterium, releases acetate, and acetate is inhibitory to bacterial growth. So there's many factors involved here, but that is why we need the oligosaccharides are so essential to establish that gut microbiome. So I just put this slide in because I just thought it was interesting to note that the effect of pasteurization on the oligosaccharides. We know that obviously pasteurization reduces the pathogen count, but it also cleaves the oligosaccharides from their binding proteins and increases their bioavailability. So potentially, as a sort of a byproduct of pasteurizing, we can also improve the commensal gut bacterial numbers. So going back to our table, there's also cytokines in their free form within colostrum. These have immunomodulatory effects and are anti-inflammatory. And then I'm just going to talk briefly about some antibacterial peptides that are in colostrum. So lysozyme destroys peptidoglycan bonds, so it causes lysis of bacterial cell walls. Lysozyme is naturally released from the Paneth cells in the small intestine, but it's also present, it's free-forming colostrum. Lactoperoxidase, it's a peroxidase enzyme. It's used for preservation of food in supermarkets. Basically produces free radicals that inhibit bacterial growth. Then lactoferrin. I just want to talk a little bit about lactoferrin because lactoferrin is used in human medicine. It's used to treat diarrhea and prevent sepsis in children. It was also used to treat patients during the COVID-19 outbreak to reduce severity of symptoms and also to treat in contacts when people were isolating. So I've got another animation here to show you. So just work with me from the top left. So lactoferrin is antimicrobial, it's anti-inflammatory, and it's immunomodulatory. If we look at the antibacterial action first, if we go to the top right, lactoferrin binds free iron. Now we know that pathogens have a high iron requirement for their multiplication, so lactoferrin binds the iron and reduces its availability to pathogens so they can't multiply. So it's bacteriostatic in that form. It also has effect on bacterial cell walls, so it's also bactericidal. Regarding what I was saying about the COVID-19, the viral treatment, it damages viral envelope proteins, and it also occupies binding sites and blocks them for viruses. So when we talk about the anti-inflammatory effects, lactoferrin binds reactive oxygen species, so it prevents cellular damage from those. It also reduces the formation of pro-inflammatory cytokines such as interleukin-1, interleukin-6, and TNF-alpha. And in terms of that one stopped, the bottom left, but the immunomodulatory effect, it promotes the maturation of T lymphocytes and B lymphocytes. So obviously those B lymphocytes will go on then to produce more antibody. So it's effective in both the innate and the acquired immune system. And as we've touched on before, the commensal gut bacteria, what happens with lactoferrin is the Bifidobacterium and Lactobacillus are low iron requiring organisms. So while the lactoferrin is taking out the pathogens because they can't, it's removing the iron to prevent pathogen multiplication, it allows the Lactobacillus and the Bifidobacterium to grow. I came across a study where lactoferrin was used to treat calves with diarrhea, and it was shown that it can reduce mortality in calves that are suffering from diarrhea. So just want to quickly run through a few studies just to back up what I've been saying. This one, we're talking about heifers and their time to first service. Two groups, one with failure of passive transfer, in fact, very, very low passive transfer, less than 5 grams per litre, and then very good, which would be over 15. Oops. You can see that the time to first service is much reduced when you have a higher transfer of passive immunity. We know then that we want heifers to calve at 24 months. If they come into the herd earlier, they've got better production when they're in the herd, they've got better fertility, they're more likely to go back and calf, and then they're less likely to be culled. So you can see the effect there. Colostrum feeding and milk production, we all know milk production is body weight. So this was a study done that showed that for each unit of IgG above 12 grams per litre, there's an 8.5 kilogram increase in milk production. It doesn't seem like much, but when you multiply it by large increases in the transfer of passive immunity and multiply it by numbers in herds, it has an effect. And then just this last study I just wanted to share with you. This is a study that was done where they just compared giving 4 litres of colostrum to giving 2 litres to 2 different groups, same quality. And you can see there that the animals that received 4 litres of first feed, their milk production was increased, the average daily gain was increased, and again, they were less likely to be culled before the second lactation. So just finally then, when we're talking about our adult phenotype, and I think this is significant to discuss because there's a lot of emphasis placed on genetics. When we're talking about our adult phenotype, what influences that? Well, the developmental programming, we obviously have the genotype, which is influential on that. But then we have epigenetics. Now, I think I've shown through this talk that one of the most influential factors in the epigenetics of a calf is colostrum management. So if we want to maximize our genetics, we need to ensure good colostrum management, because tissue plasticity in an adult animal is minimal. In a young animal, it's very susceptible to influences. So we need to intervene at that point. So what I'm trying to say is maternal signaling to the calf is not complete when it's born. There is a post-birth period where maternal signals are transferred through colostrum to influence growth and development. So if we deny the calf these, then we are definitely going to impede their development. And what does it lead to? It leads to better feed conversion, which obviously then comes into sustainability, better reproductive efficiency, better weight gains, and increase in milk production. So conclusion is good colostrum practices support the genotype to define the productive phenotype of the cow. If we want to maximize our genetics, when the calf is born, it is not fully developed, so we need to ensure good colostrum management. So just in conclusion then, as veterinary surgeons, I think we need to be aware that colostrum is nature's perfect cocktail to ensure immunity of the calf and enhance growth and development. And if we want to make a difference in terms of productivity and animal health, reducing antibiotic usage, and ensuring animal welfare, then we need to concentrate on good colostrum management. That's it.
Thanks, Dr. Davis. We have time for some question. If any question for the speaker, you can ask directly in the microphone in English, please. Some question? No question. All is clear. All is clear. Okay. Thank you very much, Dr. Ryan Davis. We give you a small present. Thank you very much.
Thank you very much.
Dr. Ainhoa, okay. Our next speaker is Dr. Ainhoa Valldecabres Inchaustegui. I don't know if the pronunciation is correct, from Ireland. Go ahead, Doctor. Yes, it's there. Okay, all right.
Thank you.
The conference of Dr. Ainhoa is Smectite supplementation to preweaned dairy calves, effects of passive transfer immunity, morbidity, and performance and mortality. Thank you
very much.
Go ahead.
Good morning, everyone. Thank you for attending this presentation. My name is Ainhoa Valldecabres. I'm a research officer in Inchaustegui, Ireland, but the work that I'm going to be presenting today was conducted during my PhD in California. So I'm going to be talking about clays, which is something that you may not be familiar directly, but you may be familiar as veterinarians in your vet practices with something called like after charcoal, things like that might ring a bell. So clays are naturally occurring fine-grained geologic materials with a history of use in healthcare practices that date back to ancient times. So they've been used in humans, and they are used a lot in different species such as pigs or poultry, but we don't have so much research or so much details of how they work in dairy calves. So that's a little bit of what we were trying to learn about when we did this study in California. So the physicochemical characteristics of the clays is what makes them usable for different applications in the fields of agriculture and medicine. We don't really know very well the mode of action of the clays, and there is only little research that has been conducted about it. But it's basically their absorbent capacity, so the ability for them to bind toxins or pathogens in the GI tract and then let them out of the system. So I don't have a fancy slide like Dr. Davis had, but you might remember the polysaccharide slide that he showed that was binding a pathogen, a bacteria, and then that pathogen left the system. So in a similar way, these clays would be able to bind toxins and pathogens in the GI tract and let them out of the system. They also have antioxidant capacities because they can exchange ions with other molecules, or those oxidant molecules, kind of protecting other cells and molecules from being the donors of those ions in that oxidation reaction. And they can also act as pH buffer. Smectites, which are the specific type of clay that we were evaluating in this research project, are among all the different type of clays, which are very variable, very different, the ones that have the larger surface area and cation exchange properties, which links them directly to the adsorbent and antioxidant capacities. So we thought that this type of clays would have higher chances of yielding a positive effect in the calves. So the hypothesis behind the work that we conducted was that supplementing newborn calves with low doses of Smectite for a short period of time will be safely improving their health and performance. The objective then of this study was to evaluate the effects of supplementing the preweaned dairy calves with Smectite on passive immunity, morbidity, performance, and mortality. The materials and methods: we conducted our study in a calf raising operation in California Central Valley, and we enrolled 200 Holstein and Jersey female calves. They were born in a nearby dairy farm, just about 1 kilometer away from the raising facility, and we just brought them in at 1 day of age and get them enrolled in our study. The treatments in our study were Smectite or control. The Smectite treatment were the calves that were force-fed 2.5 grams of Smectite mixed with 5 mL of water. That Smectite that we were using in our trial was sourced in a mine in Utah. And the reason why I mentioned this is because clays, and the one that we were using in this trial was a natural occurring clay. They vary a lot, even within the same mine. So that's why it's important to refer to what was the source of the clay that we used in this study. And it's something that is going to interfere with the repeatability of the results that we see in this study and what other studies might see. So we force-fed that paste of 2.5 grams of Smectite, and then we followed it with 50 mL of water, all administered with a syringe orally, and then we had the control calves to which we only gave the 50 mL of water. We gave the calves the Smectite and the control between the morning and afternoon milk feeding for a period of 10 days. So we enrolled the calves at day 1 of age, and then daily between the morning and afternoon feeding, we would be feeding that Smectite clay paste up to 10 days of age. Before we got the calves in our trial and they arrived to the calf raising operation, they were fed 6 liters of colostrum in 3 separate meals at the dairy farm source. And that's something that we didn't deal with. That was only done in the dairy source before we get them in our calf raising operation. The data collection: we collected different types of data for this study, blood samples to determine IgGs, total proteins, hematocrit, and minerals. We collected that blood sample as a baseline before we started the supplementation of the treatment. And again, a couple of times during the supplementation period, at day 4 and day 8. And again, a couple of times after the supplementation finished, at day 12 and 16 of age. We also measured the calves, kind of an indirect way to looking at the growth of the animals and body weight. And a researcher in our team, who was also a vet by training, was assessing the calves from day 1 to 20 of age on health parameters. Then we also relied on records from the farm for the therapeutic treatments, antimicrobials and supportive treatments, as well as for the mortality records. And we collected that type of information from day 1 to day 30 of age. The type of analysis that we used for the total proteins was a digital refractometer. IgGs were measured by RID, and the minerals by ICPOS. The statistical analysis was conducted with SAS. We used multiple linear regression models for the continuous outcomes, the blood parameters and the body measurements, and then the health parameters, therapeutic treatments, and mortality were analyzed by logistic regression. We included in the models the effect of bread, because you might remember that we had Jersey and Holstein calves, as well as hematocrit as an indirect assessment of hydration status of the animals, and baseline was also included in the models when it's statistically significant. So looking at the results of passive immunity, the Smectite calves tended to have higher serum IgGs than the control calves, and that was driven by differences that we observed mainly during the supplementation period. So back to the slide, in the y-axis, we have the IgGs in grams per liter, and in the x-axis, the time relative to calving. You may remember from the slide that Dr. Davis showed the Lombard classification. Our calves started at a good condition on IgG levels, both treatments, that's the baseline point. And then during the supplementation period, which was the samples at day 4 and 8, we see higher levels of IgG for the Smectite calves compared to the control calves. Then post-supplementation, the levels look similar for both Smectite and control calves. So as I was mentioning initially, we don't know very well what's the mode of action for the clays in this digestive tract of the calf, but there's been done some research mixing clays in the colostrum, and it's been showing that there is an increased uptake of IgGs by the calves. We were not feeding it in colostrum. You might remember we only enrolled the calves at day 1 of age, so the mechanism, I will tell you in a minute, it's probably different from what we saw in this study. But what they suggest for the calves that have an increased uptake of IgGs when the clays are fed into the colostrum is that there is, well, also again, referring to the previous presentation, if we decrease the bacterial load of the colostrum, if we decrease the potential negative impact that bacteria might have in the GI tract and absorption of IgGs through the presence of clays, by binding this bacteria, we might increase the chances of having a bigger absorption. There is also some description that this lower the passage rate that the clays might induce by making more viscous the liquid of the colostrum might also give bigger chances of absorption of IgGs, as the colostrum would spend more time in the GI tract. And that's what they suggest for the colostrum feeding. But again, we were not putting the clays in the colostrum, so that was something else rather than probably that IgG absorption. So what we were thinking is that it might go in similar lines. The fact that we might be decreasing the presence of bacteria or the absorption of toxins from the GI tract to the systemic circulation might have helped our calves to maintain the passive immunity higher during that period of time. There was a lower demand, if you will, of those IgGs to be used for a challenge. So that's what we are thinking that might explain our data rather than what was going on with the colostrum feedings, which is equally very interesting. Looking at the total protein and hematocrit, we didn't see treatment differences for total proteins or hematocrit. As you can see, for both the supplementation period and post-supplementation period, our treatments looked pretty much the same. For the minerals, the reason why we were looking at the minerals was not really to determine the absorption of the clay, which would have been interesting, but it was mainly aluminum and silicate what was present in the clay, and we were not looking at that in the bloods that we collected from the calves, but kind of an indirect way to see if there was any anti-nutritional factor in our clays, because clays are not so specific at binding. You can make them more specific if you make them chemically in the lab, but we were using a naturally occurring clay. So they might bind the pathogens, the toxins that we want them to bind, but they could also bind nutrients. So that's why we were looking at these minerals, kind of trying to see, can we see a difference in any of this? And the answer to that question was no. We didn't see a difference in these macrominerals, calcium, phosphorus, magnesium, or trace minerals such as copper, iron, and zinc. So Smectite and control calves had a similar level of these minerals in blood. Looking at the health parameters, they were measured from enrollment to day 20 of age by a research veterinarian. So we were focusing in the fecal consistency. That's also some backup of research suggesting that clays might decrease the incidence of diarrhea in calves. So that's why we were very interested, and that was kind of the main health outcome that we were looking at. So we classified the fecal scores, and here are the results for what was equal to 2 or over that. So I'm going to be presenting the results for the health parameters as the results during the supplementation period, those 10 days, and then the full evaluation period, which will be from the day that we started at day 1 to day 20. We didn't see a statistical difference in fecal consistency, so the incidence of having a fecal score of 2 or greater was similar during both the supplementation period and the evaluation period. And some limitation of the health parameters that I'm going to be showing you, and you're going to be wondering why they didn't find anything. Our study was not perfect, as most of the studies that we do. One reason for this that might have limited our ability to detect effects on health parameters was that we were evaluating the calves, between the morning and afternoon feeding, and usually in the US, treatment practices or treatment of the sick animals is going to occur after the morning milking. So that might have induced some changes in what we were measuring later compared to what they were recording early on. So milk refusals across the treatments were also similar. During the supplementation period, the overall incidence of refusals was 5.7 for Smectite, 6.1 for control. During the full evaluation period, very similar numbers again. The respiratory disease was also similar across treatments. The supplementation period, only 2 cases of respiratory disease were observed. They were doing the control calves, but we wouldn't run statistics with such a small number of animals. And during the evaluation period, the difference was not statistically different either. The body measurements, we didn't see effects of treatment in those heart breadth, body length, height at waiters, or height at hip at day 12 or 60 of age. The antibiotic treatment, something that was of high interest to us because everyone is trying to decrease the use of antibiotics in commercial farms. We didn't see the statistical significance. This was recorded by the farm employees. We only went through their books and then extracted the information and analyzed it ourselves. And that was from day 1 to day 30 of age. So during the supplementation period, the prevalence of calves that got at least 1 treatment of antimicrobials was 5% for Smectite and 8% for control calves. And then during the full evaluation period, 28% for Smectite and 25% for control calves. The supportive treatment, all that supportive treatment that it was not an antibiotic, was also similar across treatments. We only had 6 supportive treatments during the supplementation period, so again, no statistics running this outcome. And then for the evaluation period, it was 7% for Smectite and 9% for control. The mortality, that's the last result that I'm going to present to you today. We observed a tendency for a lower mortality in the Smectite calves. So that's a very high, quite high p-value, but we have to keep in mind that in the end, the number of animals that died during the project was only 11. Interestingly, 2 calves died during the supplementation period, but when we look at the full evaluation period, those 30 days from day 1 to 30, we saw that Smectite calves, like there was a 3% incidence of mortality compared to 8% for control calves, and that's what this graph is plotting out, like the 3% mortality for Smectite and higher percent of mortality for control calves. Like, we didn't have a small study in the sense that we had 200 calves, but you know that sometimes p-values are just the result of how many animals do you have in your trial. If we had double number of animals, most likely other differences that were not statistically significant at this point would have been statistically significant. So this is very much like preliminary research, trying to encourage future research. We see this very positive from the sense of it warranting further investigation. Clays have been proved successful with other species. They've been used in humans for a very long time, and we are very much convinced that it is worthy to keep looking at the use of clays in pre-weaned dairy calves. So, in conclusion, our research suggests that supplementing newborn dairy calves with Smectite from a mine in Utah did not have negative effects on health and performance, also something important to clarify, it seemed to be safe, and might have positive impacts on maintenance of passive immunity during the supplementation and on mortality up to 1 month of age. Our findings call for more investigation to confirm the safety and efficacy of Smectite supplementation in newborn calves, to determine the most effective course of treatment. What we selected to do in our trial might have not been the optimum approach, and to identify risk population that might benefit the most from giving that clays. And thank you very much for your attention.
Thank you very much. Any question for the speaker? Yes, you can use the microphone. Here, directly in the microphone, please.
My name is Bednarek from Poland, from National Veterinary Research Institute in Puławy. My question is, it's on the European pharmaceutical market is successfully used in human medicine commercial product which called Smecta. I don't know, it's the background of the use of the product in human medicine against the diarrhea in different groups of age of group of people. So the background is the same, maybe similar use in calves, because your results are very interesting, especially if you context the increased, in the significant increased passive immunity if you talk about IgG concentration in sera. But I don't know what is the results of the use the product in human. Probably is the background, maybe is a protect background to the intestinal microbiome and microflora. What's your comment about the use and eventually the potential shredding of the wider use the product in veterinary medicine too?
So if your question is regarding the use of clays in animals, not humans, I assume? So I see like the limitation of the clays is the huge variety that we have. So we need to characterize very well which clays work, which ones are safe, and trying to learn more about the mode of action of each clays. But as you were mentioning, like I don't see, what we didn't see in our study and we haven't seen in the literature are negative impacts of clays. If you were to feed a very high dose, there might be impactation and constipation, but that's not something that at the doses that we are using we observed. So I would be advising like towards the use of clays as they've seen as safe. Like the EFSA in Europe recommends a 2% dry matter addition of this type of clays as safe, because there is no potential of being harmful effects associated to it. Protecting the mucous barrier by protecting from the action of bacteria and pathogens that are in the digestive tract, it's also been reported. It's been reported that there is more mucus in the GI tract of those animals that are fed with this glaze. So there is definitely potential positive effects in these calves that perform as monogastrics at this point.
Eventually, this direction will be used in praxis, in the vet praxis. I think it's maybe a little different mechanism like in human, because it's a veterinary. It's completely another process, and the calves after the colostrum passive transfer. The conditions inside the intestine and membrane intestine complete another in the human during the diarrhea. Thank you very much.
Thank you. Thank you for your comment.
Okay. Other question. Go ahead.
You were jogging my memory there. We used to use and I'm frantically Googling, sodium bentonite.
Okay.
Is that the same as Smectite or similar?
Smectite is a type of bentonite.
Right.
And the sodium bentonite, it's likely going to be a bentonite that has been modified to have more sodium ions in the surface. So that's kind of to enhance that ion exchange or that absorbent capacity of that bentonite.
Yeah. So we did anecdotally see effects in New Zealand dairy calves by top dressing calf concentrate with sodium bentonite. And the calves would have little white muzzles from eating the sodium bentonite but obviously they probably were not measuring it, and it was anecdotal, but it's interesting that you were seeing some mortality benefits of the...
Did you see any effect on health?
Yeah, anecdotally, we never studied it, but maybe it's an opening,
Yeah, we heard about it, and we are just trying to report it to get some evidence behind it. Thank you.
Okay, thank you very much, Dr. Inchaustegui. Thank you very much. Our next speaker is Dr. Kristen Edwards. Okay, go ahead. Thank you, Dr. Kristen Edwards. with a conference, Evaluation of Bovine Colostrum Replacer Supplementation to Improve Weaning Transition in Holstein Dairy Calves. Go ahead. Thank you.
Good morning, and thank you for the introduction. Again, my name is Kristen Edwards. I'm a bovine veterinarian as well as a PhD candidate at the University of Guelph in Ontario, Canada. And I'm really excited to be here this morning to share my research on the evaluation of supplementing bovine colostrum replacer to improve the weaning transition in Holstein dairy calves. Weaning involves the transition from a liquid to a solid diet, and it results in considerable modifications to the gastrointestinal tract, as both the primary location for digestion and the primary energy source change during this time. And unfortunately, dairy calves often fail to consume enough solid feed before weaning. And this has been shown to result in increased gastrointestinal epithelial permeability, so in other words, leaky gut, reduced immune function of the gastrointestinal tract epithelium, as well as morphological changes to the villi and crypts of those intestines. And as a result of these stressors, calves often experience reduced weight gain during weaning. And this has important economic ramifications, as it's been shown that for every additional 100 grams per day of average daily gain from weaning until breeding, there's an associated additional 820 kilos of milk across the first 3 lactations. So if we can improve this weaning transition, we have the opportunity to improve future production. And one potential mechanism for helping ease this transition may lie in the use of bovine colostrum replacer. Bovine colostrum contains a myriad of bioactive molecules, including growth factors and antioxidants that are involved in the immune, metabolic, and endocrine pathways. And actually, in weaned piglets, the use of bovine colostrum replacer has actually been shown to mitigate some of those negative effects of weaning. And specifically, in weaned piglets who were supplemented with bovine colostrum replacer, they had increased villus height, reduced epithelial permeability, an improved feed intake gain, and an improved feed to gain ratio. So that sort of led us to the objectives for this study, which was to evaluate the effect of supplementing bovine colostrum replacer during weaning on intestinal permeability, as well as weight gain in female Holstein dairy calves. And so we had 2 hypotheses. We felt that calves that were supplemented with bovine colostrum replacer would have reduced intestinal epithelial permeability, as well as improved average daily gain during the weaning period. And so what we did for this study was we enrolled 65 female Holstein dairy calves on a commercial dairy farm, and they were housed individually from birth until 70 days of age. They were fed a milk replacer 3 times daily, up to a total maximum allotment of 12 liters per day, and that was fed until 56 days of age. At which point, on day 57, they began to be weaned, and they were weaned over 8 days, so weaning was completed on day 64. And the way that these calves were weaned was they went from the 3 times a day feeding down to 2 times a day, and they were fed twice daily from day 57 to 60, and then they were further reduced to just once daily feeding from day 61 until 64. And so on the day of weaning, so on day 57, those calves were randomly blocked by their birth weight, and they were enrolled into one of 2 treatments, which were equal in metabolizable energy, and they were fed once daily during weaning, so from day 57 until 64. So the first group was the control calves, and there were 31 calves in that group, and they were fed 3.8 liters of the same milk replacer prior to weaning at the same concentration, so 150 grams per liter. And then in our colostrum supplementation group, we had 34 calves, and they were fed a blend of milk replacer and bovine colostrum replacer. So more specifically, that bovine colostrum replacer was reconstituted at a concentration of 125 grams per liter, and 950 mils of that was fed, mixed with 2.85 liters of their milk replacer at the standard concentration of 150 grams per liter for, again, a total volume of 3.8 liters. And so I'll briefly describe our experimental timeline here. So at birth, all calves were weighed using an electronic weight scale. And on day 2 of life, so at 48 hours, they were bled for serum IgG. We monitored daily individual starter intake on these calves 1 week prior to weaning, so starting at day 50, all the way up to 1 week after weaning was completed, so day 70. And so as a reminder, they were weaned from day 57 to 64, so that's that blue rectangle. We also respiratory scored them daily using the UC Davis BRD scoring app. And then to address one of our hypotheses, we assessed these calves for intestinal permeability on the day prior to the initiation of weaning and on the day after weaning was completed. We also assessed these calves for lung consolidation using thoracic ultrasound by a single trained observer. And that was performed the start of weaning, the end of weaning, and then 1 week after weaning was completed. And then at these same time points, we bled the calves for serum beta-hydroxybutyrate. We also examined these calves daily for fecal consistency, and we started on day 57, and we did that out to day 70. And so on day 70, these individually housed calves left their individual pens, and they were moved into group housing. Each group consisted of 10 to 12 calves, and each group was a mix of both the control and the colostrum supplemented calves. So what did we find? Well, we were unable to detect any differences between the treatment groups in terms of intestinal permeability, lung consolidation or respiratory score, fecal score, or morbidity. And so as a note, I forgot to mention, we did monitor morbidity from birth all the way out to 84 days. Same with mortality. We had no deaths, so we were able to follow all 65 calves. We were also unable to detect any differences in terms of starter intake from day 50 to day 70, so while they were individually housed. But the story doesn't end there. What was really interesting and really exciting is that despite not finding any differences in those variables that I just mentioned, we were able to detect differences in gain. And specifically, at day 77 and 84, the calves that were fed the bovine colostrum supplementation were 2.79 and 2.76 kilos heavier than the control group. So those gains that we observed occurred 2 and 3 weeks after weaning was completed, and also 1 and 2 weeks after they entered group housing. And so this could have really important economic ramifications. Again, with this kind of gain, we may be able to achieve 820 kilos of additional milk across those first 3 lactations, which on the Canadian milk blend price of $93 a hectoliter, that's approximately $760 of additional revenue. And on a US price of $20 a hundredweight, that's about $360 US dollars of additional revenue. And so, of course, there's a cost, but for each calf to be fed this colostrum supplementation, it was $50 for the entire 8 days. So there's quite a return there. There were, of course, some limitations to our study. The first being that we did have a short and abrupt weaning of 8 days, so our results may not be applicable in situations where calves are weaned over a more gradual and stepwise manner, which is how we recommend to wean calves. But nonetheless, a meaningful number of dairy farms still practice a fairly short and abrupt weaning period, so this may be a tool that could help those dairies. As well, although we did control for metabolizable energy, we did not control for fat or protein, so it's possible that that contributed to the differences we saw in gain. However, I do think it's important to note that in previous studies that have examined the effect of feeding additional fat or protein, any differences that were observed during the feeding period, which contrasts to our study where we saw the gain benefits 2 and 3 weeks after the feeding period had finished. We also were not able to elucidate the mechanism of action, so this would be important for future research. And lastly, as this is the first study that's looked at using bovine colostrum replacer for weaning, we don't know what the optimal dose is. So it may actually be less than what we used, which would actually just improve the return on investment. So with that, I'd like to thank you for your attention, and I'd certainly like to thank everyone involved in this. First, my PhD committee. So at the University of Guelph, Drs. Dave Renaud, Stephen LeBlanc, Trevor DeVries, and Mike Steele, and at the University of Vermont, Joao Costa. I'd also really like to thank the Saskatoon Colostrum Company for their support in this project, along with the dairy farmers of Canada and NCIRB. And then last, but certainly not least, I would really like to thank Sylvia Bell, who was the calf manager and is the calf manager at this dairy farm, who worked every single day for 3 months straight to ensure that this study was done correctly. So thank you for your attention. And with that, I'll take any questions.
Thank you, Kristen Edwards. Any question for the speaker? We have time. Okay. No? No question? Okay, thank you very much, Dr. Kristen Edwards. Thank you very much. You are the next speaker? All right. Well, it's thinking we have 10 minutes before the... no, but... no. Yes. Go ahead. Well, our next speaker is Dr. Kenneth Krogh from Denmark. Welcome, Dr. Krogh. The conference is description of different feeding strategies of transitional meal of 5 Danish cattle farms. Okay, go ahead. Thank you. Thank you, Dr. Krogh.
Hi, everybody. This is not a deep scientific presentation, but it is a description of different feeding strategies for transition milk. Actually, I'll start to tell who is behind this small project, because this is an informal cooperation with some parts who has a passion of a good start of the calf. It's some MSD people working with veterinary products. It's a company from Denmark called Calves who is making solutions for colostrum feeding. And then it's veterinary practice, LVK, with 22 vets. And actually it was Lene who supposed to be here, but she's high pregnant, so I'll try to do it. Okay. Let's see. Everybody knows that colostrum feeding is essential for immunization of the newborn calf. We had a good presentation for a start this morning, and the focus has been in many years to make the first 4 liters quick as possible, within 4 hours or something like that. After this first colostrum feeding, the calf typically will not be fed by colostrum or transition milk in many cases because of practical issues. There's been very much focus on the first time, and many farmers know it's important and try to set up systems, but in many places they don't use the transition milk during the first days of the calf. And I think it has something to do with there's not very good practical solutions for this, and they don't get the good things. As we have heard here from the beginning, there's a lot of good things in this colostrum who can support the gut in the first days after the raw milk. So also there is a lot of IgA in this transition milk, who has to get local immunization, and even when you are going to vaccinate, you have the specific IgA. So the focus here on this project was to, the object was to make this study to find out of practical solutions to support some farmers who was interested to start to feed transition milk. We can say it in another way, to come from theory to practice, and how can we help them to support the system so they get it done. 5 farms were motivated here. We took them together in a group and discussed with them what our theory is, and we knew they were interested in starting to join the project and start to find a solution for using this transition milk. The 5 farms were quite big farms from Danish purpose. There were 2,200 milking cows. And before and during the projects, we were supporting them by some measurements. It was especially IgG measurements to check blood checking of immunization. We also do some bacterial counts, because one of the big issues here was that when they find different solutions, they keep the milk longer and cooling and so on. So we have to be sure the hygiene was okay, so we didn't get worse and infection and these things. And then we knew that this could perhaps have an impact on the diarrhea. Many farms, if you are going on looking for diarrheas, they have a lot of small diarrheas, and we think this was one of the ways of helping with that lab problem. So we had also a diarrhea scoring system we were using. To support this practical aspect, the focus was on different methods of collecting, storage, and feeding transition milk. I'll show here some of the measurements. To the left, this is blood samples where we are taking 10 blood samples and looking with Brecht's method for serum, and we use the same scale as which many are using during these years in 4 categories, and we put it here. We can see the very good. Unfortunately, this is in Danish, but very good is green. There are 2 of the 10 who is very good. There's no good, but there are about 6 here who is fair, and 1 bad. So actually, this is not a quite good example of passive immune transfer. Another one we were using very much was to check hygiene in different situations. With bacterial count, we have a PCR method where we can, with CT values for different bacteria, you can see here Bacillus, Clostridium, Streptococcus, Pseudomonas, and Enterobacteriaceae. This is the 4 groups this test is taking, and then we can transfer the CT value to CFU. Here's 3 examples. In the middle, that is one taken immediately from the freezer. There's a little bit, but not very high, a CFU. There is one to the left, 2 days old milk stored in a fridge. There's coming some Enterobacteriaceae growing there. We can see earlier we would like it to be under 100,000. Here it's more than 250,000. To the right we have another example where we have checked the milk exactly from a bottle where they are suckling it, and this is also high. Just some examples of using this tool to check. This is a very simple scoring system. Everybody can do it. We said you have to put them in 3 categories. If it's a normal feces, it's like pudding. If it's a mild case, it's like pancake dog, and if it's a severe case, it's like orange juice. Everybody taking care of the calf can do that and put a mark on a paper, and we can make some calculations. Here is the scheme of the 5 farms, what was happening when we were doing this. Yes, we can see all here, all the farms here, 5 farms. They all collect milk from the first milkings. Most of them from the 2 and 3 milkings after, and 1 up to 4 milkings. Here is 1 collecting milk up to 6 milkings. And they all pooled the milk getting out of this, but they had different systems of pooling it and storing it. And we can see here, and storing procedure, somebody started to have a combination of freezing and cooling. But later on, many of them find out it's more complicated with the freezing system, but it can be a good backup to have it. So cooling was the most often one. We have 2 here who have no storage, quite big farms. They fed it fresh. And there was different kind of storing equipment, and I will show some pictures just after this here, because there are here both cooling tank, freezer, and fridge. Here there are milking tanks and fridge and a cooling tank, and then there also are, perhaps you also know it, the Danish system Colostrum, made in Denmark from calves. They have this colostrum bag system, and that is also can be used to cool and to freeze the thing in. There's one here who made a special wagon with a separation, and the feeding procedure was also different. Some of them think it was important to give this, so to be sure they took it, so they fed it separately. And when I drank this, then they supplemented with the milk they used, and that could be different things. One was using milk replacer, some were mixing with waste milk and such things here. You can also see here there was different volume. This system here, they said, okay, we'll in 3 days make 1 liter of this good stuff, once a day. One over here, 2 of them say we will do it twice a day and using 200 to 500 mils. But they did it in 14 days. We were not telling them what to do. We were telling them that it was important to do it in some days. And here there's one making 4 liters and 2 times per day in 5 days. And this last one here actually was taking milk from the first up to 6 milkings. So there was quite a lot of milk here they have, because many of them didn't take up to 6 milkings. But they pooled it with the waste milk. And you can say, was that much different than doing it as many others are doing it today? But when we calculate it here, we try to calculate it back, so they actually, because they have all these milkings, they were using up to 100 to 300 mils per day. So that was the difference here. Okay? And here is some of the some pictures from some of the situations. It's mainly here we can, oh, we'll take it back. We can see here is the Quick colostrum bags containing plastic bags with 4 liters. It could be used to storage it. Actually, I could hope that they will make the system so they could easier get 1 liter out or something like that, because then they can use it better in this situation. Here is just a normal fridge and a system where they try to put in buckets and put date on, so they have a system to be sure that they are not getting too old here. And that was also why we were going to make some of the measurements of the bacterial count. Here to the right we have some systems. Of course we have this Quick system here, and we have just a fridge to put in. But this kind of tank, it was a 200 liters tank where they can cool it down. Actually one was buying it, and another farm said, "Where have you got that?" Because that was very, very good, he said, because we see a lot of situations where they take the transition milk, put it in buckets and everything, and the hygiene is not good. So to have a system here, if you have to prioritize this and really do this, you should have a system and a separate cooling tank for that. So actually you can buy these small tanks. Here is some of the feeding systems. Here we have Farm 1 using a milking taxi, and actually they were very fit. When she got the idea, I said, "I'll buy a lot of 1 liters of marmalade buckets." She found a place to buy them, because she said they should be clean, we'll use a clean one every time and so on, and they got them cheap. And she started to have 1 liters in that, and she tried to put out here to see when she was going around with the milk taxi, she could have this 1 liter bucket and put it in for every calf every day, once a day. That was her solution. Here we have this self-made wagon where you have the normal milk you are giving, but they have a small separate room here where you could put the transition milk in. Very simple system, but you can't buy them, you have to do something yourself. Here is some of the few results. We had 3 of the farms were making this scoring of diarrhea, and Farm 2 here had a quite high level of mild and severe cases of diarrhea, and the motivation was that they should do that before, and then should start with the system and see if there was an impact. And actually this was very important for them to see, and you can see all 3 farms had a decrease in the numbers of mild and severe diarrhea. This was decreased by 46%, also this, but from a small level, and from a level in between, they nearly disappeared. Of course, this is just a moment picture of what we are doing, but it was very important for them to see that it helped in some way. Actually, we had one farmer here, she was very enthusiastic about this. You probably can imagine here, she has a long paper. She took all the calf, had them every calf and every day making a scoring for diarrhea. She even put them in with different colors. So if it is a severe case, it was more red or more yellow. So you can see here, probably see here in the beginning, there's a lot of yellow things. There's not so many here. There's coming something here. And she even can see eyes starting on day 2 and 3 and so. It was important for her to see, but also to show to the all other calf takers to motivate them that what they are doing will help them, because it is more work and more complicated. Okay, we are coming to the end here. The take-home message here from this practical trial, or what we can call it, is that if we should do that, we have to be sure that we don't miss something, and therefore it is important that we try to support with some KPIs. We used IgG on serum, we used diarrhea score, very simple system as you can see. Some bacterial measurement is very important also. And it's important that the farmers can see it will help. Therefore, and we showed here in in 3 of the farms who actually did this scoring, that there was a positive impact on diarrhea score. But overall conclusion, I will say there is that transition milk feeding demands a practical system and some equipment to be practiced. And I don't see them out there, these systems. They have to do it and set it up themselves. And therefore, I think it's very important that we as veterinary advisors and consultants should be better to advise and inform on the benefit of transition milk feeding, and perhaps showing them some practical solutions to set it up. That was the purpose for this. Thank you.
Thanks, Dr. Krogh. Any question for the speaker? Yes, go ahead.
Hello, thank you very much for a very interesting overview of the Danish sector. I was wondering, maybe I missed this piece, but did those 5 farms never fed transition milk before you gave this overview or introduction about transition milk feeding for them? Or did they feed or did something and then they changed something after you instructed them on the best practices?
I think there was one farm who was starting on it, and some of them were interested in doing something, had an idea about something to do. They had discussed it together with the vets and some from this here. So it was not just just before and just after. They had an idea, because we had to choose some persons. We said we will make a group of this person who is interested. So we have to choose some who is willing to start to do something new and show others how to do it.
Okay, thank you.
Okay.
Thank you. Another question? No? Okay, thank you. Thank you very much, Dr. Krogh. Now we have a break. See you into the room at 11 o'clock, please, for the second keynote lecture. Thank you. Hello, everyone. We invite you to take your seat to continue the program of the nutritional section. Okay, now we have our second keynote conference. The speaker is Dr. Juliana Leao from Brazil. Dr. Juliana Leao, based in Wageningen, the Netherlands, grew up in the largest milk producer state of Brazil, Minas Gerais, and graduated as a veterinarian at the Federal University of Minas Gerais, Brazil in 2011, where she was part of an extension program to improve milk quality and reduce mastitis in dairy herds. In 1910, started working with dairy calves in the lab of Dr. Sandra Gesteira Coelho. Juliana then earned her Master's Science 2013 and PhD in 2018 in Animal Science from the same institution, focused on neonatal calf nutrition and management. Juliana spent 1 year of her PhD in Embrapa Gado de Leite Coronel Pacheco, Brazil, focused on calf feed efficiency, residual feed intake, thermography, and energy expenditure. After her postgraduate, she moved with her family to the Netherlands and started working from SCCL as a technical specialist assisting different countries and clients on colostrum management and application of colostrum. The conference of Dr. Juliana is Importance of Colostrum for Small Ruminants. Thank you very much, Dr. Juliana.
Is this working? Yeah. Thank you so much for this kind introduction. It's a pleasure to be here today. I asked to have this microphone. If you're not hearing me well, I can go back to this one. So you tell me, okay? So before I start, first I have to tell you that I'm very, very proud and honored to be here today. I have an excellent time just talking about colostrum all the time, and I'm happy that you are here today so we can discuss a little bit about colostrum in small ruminants. So I do like to read a lot of research and papers, but for me it's super important when I'm reading something that I can go back to the farms and to my clients with something that I can apply on farm and give them a solution for their problem. So today I will try to gather all the information that we can get off this room to your farms, your clients, and change something on farm that will make an impact. Is that a deal? Okay, great. Before I start, I would like to know who here is from Mexico? Just raise your hands. South America, New Zealand and Australia, UK, Europe in general. Great. Very good. Because then we can go out and apply some science, okay? Okay. Some rates that are important to talk about is neonatal mortality and why colostrum is so important to impact on neonatal mortality. We know that those rates of 15% of mortality are not changing for the past 40 years. In one side is good, because we vets can work for a long time trying to tackle this mortality. In the other side is not so good. We are not doing a good job. We need to change some things on management to change these mortality rates. Most of this mortality, 50% of this mortality, will occur on the first day after birth, and the major causes that these animals are dying is because they are hungry, so they are starving, they are too cold or too hot, they are not doing thermoregulation, they are in risk of any disease. I don't know where they are born, maybe in a barn, very dirty, full of E. coli, and then they have watery mouth in the UK, for example. So we need to tackle these causes of mortality. We also know that around 10 to 30% of the animals do not produce enough good quality colostrum. So when the neonatal losses occur, as I said, half of this mortality occurs at birth, on the first day, and some others between scanning or on the first 14 days. And we are going to talk about colostrum management. So we are going to talk about this mortality on the first day that they are born. Oops, I'm pressing the wrong buttons, guys, sorry. A lot was discussed on the first talks today. Dr. Ryan explained very well what are the components of colostrum and how each component play an important role on the neonatal survival. And I cannot talk about colostrum management without talking about early life nutrition. Oops, guys, this is not working with me. Early life nutrition. We know that early life nutrition in dairy calves, they impact average daily gain, they impact health status, and they impact survival rates. We know that a calf that gains 1 kilo in the pre-weaning period, they will become a cow that produces more milk in the future. And the same will be for small ruminants. We also know that if we lack of giving a good colostrum, these animals are going to be at great risk of getting any disease, so they can die, right? How can we tackle this mortality issue? By having a good colostrum management. We need to overcome starvation, hypothermia, birth trauma, and infectious disease. And with that, I will go through with you this colostrum basics. So this presentation, when I'm talking here, you might think, oh, it's too basic, I don't need that. But if you go back on farm, I'm sure if you look at how quick you are providing colostrum to your animals, or how is the quality of the colostrum, what's the quantity that you are giving to the animal, to the goat kid or to the lamb, how clean is my colostrum, and how I quantify the passive transfer of immunity, I'm sure you will find something that you can improve. So what's colostrum? It's the first mammary gland. Oh, it's the first mammary gland secretion that the ruminants produce, and it's super important because they are born with naive immune system. It's essential for their development. It's essential for them to survive. It's rich in IgG, it's rich in fat, it's rich in oligosaccharides, and each component plays an important role for the survival of the animal. What is the importance? First, nutrition. These animals are born, they are very light, they need to survive, they need to thermoregulate because they are inside the mom, the dam, and then they are outside. Is the temperature going to be the same? Is the temperature going to be the same? No. They need to thermoregulate. They also need colostrum because, as I said, they don't have any immunity. They need to acquire immunoglobulins to acquire the transfer of passive immunity to not get sick. They need to maintain their body weight. They need to make the bond mother and offspring. And also, there are growth factors that are important for the maturation of the gut of the newborn. And it's important for microbiota colonization, like Dr. Ryan explained on the first talk today. Super important. Impacts of colostrum. By giving a good colostrum protocol, you will help with the ability to survive, you will improve production, you will help with the gut development and maturation of the gut, And you will help reproductive efficiency, because these animals are going to gain more weight, they're going to reach the reproductive time earlier, and you're going to get more milk or meat earlier than if you are not giving a good colostrum management. So the components of colostrum of small ruminants, we have a very high percentage of fat, percentage of protein that is lower than in bovine, and I'm going to show you some graphs that you can see that. And it's very low in lactose. But colostrum also gives an energy of 6 to 7 kilojoules per ml. And this information is going to be needed because we are going to do a small calculation. Who here likes some mathematics? I hope everybody. If not, you just take a picture, and when you read again, you will understand what I'm trying to say. So like Dr. Ryan explained, colostrum also has oligosaccharides. They are very important because they are like a prebiotic for bifidum bacteria. Bifidum bacteria and lactobacillus are super good bacteria, and as we know, good healthy gut, healthy life. So we want good bacteria to be present in the gut of these small ruminants. Oligosaccharides also work as bacteroids, so they bind to bacteroids and they impair the colonization of bad bacteria in the gut. And they also bind to bacteria like E. coli, again, UK, New Zealand, Australia, super important. Oligosaccharides also help on the uptake of immunoglobulins. Okay, what is that? What are you saying? I'm saying that if you give colostrum on the right amount, good quality, as soon as possible, these animals are going to be more able to absorb more immunoglobulins, to have a higher transfer of passive immunity, and to be protected for longer. It's very interesting, right? Okay, colostrum composition and different species. We have here this study that was performed in 2019 from Switzerland, and they compared bovine colostrum, goat, and sheep, and as you can see, the composition is very different. The IgG in bovine is higher than in goat and ovine, but the fat, as expected, for the ovine is going to be higher than the goat and bovine, and the protein also higher in sheep and lower for bovine and even lower for goat. Okay, but what if I want to use one species colostrum in the other species, if I don't have colostrum? You can, because there is an homology between IgGs, between species, so it's okay, you can. About the composition variability, this study, very interesting, super new, was published in March this year from a group of China. And the blue line is the colostrum composition, and the orange line is the milk composition. We are only going to talk about the first three, so cow, goat, and sheep. And as you can see, the dry matter is higher on the cow, lower on the goat, and even higher with the sheep, because, as we saw on the slide, the previous slide, higher fat, higher protein. So here, higher fat on the sheep, higher protein, sorry, and higher fat on the sheep. Lactose content, low in all the species. So let's focus on immunoglobulins, IgG, because I want to talk about transfer of passive immunity. And we are going to talk about only one species so you can understand where I want to get to the point. So this study, also from this university, this group of researchers, sorry, and they got goat colostrum, dairy goat, and meat purpose goat. As you can see, the concentration of IgG varies drastically depending on the breed, and it also varies between the sheep. Sorry, I said ovine, right? It's sheep. Okay, it's different. So how can I know what my newborn needs if I have this variability between species, between breeds? How can I do that? How can I have a protocol? It's very difficult. And no, it's not. It can be very simple. We just need to understand how we can have different colostrums independent on the system that you have. We know that herd management will affect, body scores will affect colostrum quality, dry period length. If you have a very short dry period length, you will have bad colostrum. Parity will affect colostrum. Breed, as I showed in the previous slide, will affect colostrum. Number of offsprings, we know that goat that has twins, they have better IgG concentration, but if you have triplets, you have higher dilution of this colostrum, so the IgG is going to be diluted, so you get a higher volume, but a worse quality. Season will play an important role as the same as photo period. So we are not going to discuss that today, because then I would need 2 more hours, and I think you would be always sleeping by the end of it. So I'm just going to focus on colostrum protocol. Passive transfer of immunity, we all know how it occurs. The goat or the lamb is born, they are outside, they need some colostrum because they need to stand up, they need to be able to suckle colostrum from the mom or from a bottle. They need to acquire immunoglobulins for the transfer of passive immunity, so they can absorb all the immunity as soon as possible. Yeah. Remember that Dr. Ryan said that as soon as possible, because then their ability to absorb will decay over time. Yes? And when we give colostrum as quickly as we can, we are improving their ability of absorbing IgG, and the higher is the concentration of IgG in the colostrum that we give, for longer they will be protected. There is a study using bovine colostrum with parainfluenza antibodies in it, and they wanted to understand how was the persistency of these parainfluenza antibodies in the bovine colostrum. And when we talk about passive immunity, we always say, they will be protected for some weeks, 3 weeks, 5 weeks, but we learn over time that if you are giving the right dose, the right quality, and as soon as possible, they can be protected for much longer. So that's very important when you are vaccinating your dams to use the colostrum as soon as possible. Okay. Remember when I said about thermoregulation, newborn adaptations? So these animals are going to be inside their dam, they are comfortable, and then they are out, and they need to thermoregulate. We have a big problem I live in the Netherlands, right? So 9 months of the year, I'm there, and it's raining. The UK is the same. And these animals, they need to adapt to this cold weather. If they are born indoor, you have a very high environmental challenge. You can have very high failure of passive transfer rates, because you have to give colostrum artificially if you are not living with them. You can have lack of colostrum because you need to collect colostrum to give to these animals. You can have poor microbiological quality of this colostrum because you have to collect. Remember the bucket that he showed on the colostrum bucket that Ryan showed on the first talk? Can be very dirty, so very high bacterial count. And it can also have poor immunological quality. If you are lambing outside, you can have low temperature, it's super cold. You are not taking any measurement on how much these animals are drinking, so you don't know they are suckling. You can have lack of energy, and they will be starving. They will be not thermoregulating, so they can have hypothermia and they can die. So risk factors of failure of passive transfer: low birth weight, We know that very small goat kids and lambs below 2 kilos, they are very difficult to survive. Dystocia birth, we know that animals that suffer from dystocia birth, they are less able to absorb immunoglobulins. They lose their ability in about 20%, which is a lot. Adverse weather conditions, inadequate colostrum supply, and competition between siblings, if you are raising these animals with their dam. Imagine if she has triplets. Do you think she will have enough colostrum for all the animals? I don't think so. And poor expression of maternal behavior. So now we are going into the colostrum protocol. Everybody's awake there? I hope so. If I ask you to grab a piece of paper and draw me a lamb, do you think you are going to draw the same lamb as the person that is sitting next to you? No. That's why we need to have protocols. So then you can standardize what you are going to do on farm. I wish we had more time so we could really draw some lambs and compare to the colleague sitting next to you. Unfortunately, I don't have. So first, I'm going to talk about time and why it's important. We know that apparent efficiency of absorption in sheep and goats is between 30 to 40%. It's not high, it's low. So we really need to focus on colostrum quality. So these animals are going to be having more chance to absorb. This is a study from the bovine world, but here shows why it's important to give colostrum as soon as possible. Because if you don't, they are going to lick the barn, lick the sibling, and they will have E. coli in their small intestines instead of IgG. Another bovine study about delayed colostrum feeding. So here we have IgG, serum IgG, and hours after birth. So they were divided in 3 groups. The blue line group received colostrum at 0 hours, and the 6 and 12 hours received at 6 and 12 hours. So all these animals received the same amount of colostrum. And look, how is the IgG concentration of the 0 hours? It's higher at its peak and persisted higher. So the 6 and the 12 hours did not achieve the 0 hour group. So we cannot delay the first colostrum. Second thing that we are going to talk about is quantity. So the mass of IgG required for a small ruminant to achieve a good transfer of passive immunity is 30 grams of IgG. This is mass of IgG. How can I give this mass of IgG in 2 or 3 meals? I need to give to my animal minimum of 15% of body weight in colostrum. Ideally, and with my clients, my farms, I give 20%. Why? Because more colostrum is better for them. That's why. I don't want to reach the minimum point. I want to be good. I want to do good for them. I want them to survive. I don't want to go back on farm to treat animals. I don't want to lose animals. I don't want to be in the 15% rate of mortality. So that's the minimum, and the 20 is the ideal. If you can give more, good. So the first meal, usually we give a bottle of 200 ml, and this in 200 ml should have 10 grams of IgG. So if you are giving 3 bottles, you are giving the total of 30 grams of IgG. If you are giving bigger bottles, good. If you want to give 10% of body weight on the first feed and then give a second feed of 5 and another one of 5, even better, okay? So remember when I said I hope you like a little bit of calculations? So let's follow this with me. Please don't sleep, guys, because if you sleep, I sleep. If I see someone snoring, I also sleep, so help me out, okay? So what are the newborn energy requirements? We will take as an example a 4-kilo lamb, okay? When you calculate the metabolic weight of this animal, which is the body weight, metabolic weight, we have this number here. The colostrum energy content is between 6 to 7 kilojoules per ml. Remember that number. So if I have an indoor lamb, they will need 1.27 megajoules per day. And if I have an outdoor lamb, which will need a little bit more energy, and if they are in the UK, these calculations is probably wrong, sorry, because it's cold. If they are in New Zealand, South Island, probably the calculation needs to be redone. They will need 1.56 megajoule per day. So if I am giving 15% of body weight, I am giving 3.9 megajoules per day. But I'm giving a first meal of 200 ml, I will give 1.3 megajoules in a bottle of colostrum. So the needs, 1.27, what I'm giving, 1.3. This is just to survive, okay? Maintenance. Now, different situations of thermal stress. Let's say that we are considering a day of 5 Celsius, and I should have done that 40 Celsius, right? Because we are in Mexico and it's very hot here. So next time I will do for hot weather, okay? So this time I considered a 5 degrees Celsius, and it increases the energy requirement in 0.25 if you are indoor and 0.31 if you are outdoor. So the amount of megajoules that they need just to survive will also increase. Okay, I have the thermal stress here, the calculations of thermal stress, but what if I have wind or rain? Then the energy requirements will also increase. They will increase again. So we will have a 0.39 megajoules need for the indoor animals and 0.47 for the outdoor animals. What if we had an E. coli issue? Are they going to need more energy to fight disease? Yes, but I didn't do this calculation. So for the weather, thermal stress and amount of energy needed for wind and wet, for the indoor animals, just for maintenance, is 234 ml of colostrum. So the bottle of 200 ml, it's not going to be enough. So let's take the bovine dairy world as an example to not get our bars of need so low. Let's aim high. Let's have better animals. Then we can improve the mortality rate. Okay? All good with the calculations? Everybody understood why I did that? Because we don't talk about energy, and it's super important. So now, moving on to immunological quality, which we talk about it a lot, and I gathered all the papers that I had available to talk about immunological quality of the colostrum. So here I have EU colostrum in Brix, EU colostrum in IgG concentration, goat colostrum in Brix, and goat colostrum in IgG concentration. So I am aiming for the good or excellent, whatever you want to call, above 26%, above 25% Brix. Brix, it's super easy to use. You probably use that every day with your clients. Has a good correlation with concentration of IgG in colostrum. But again, me as a good vet, but daughters of engineers, I like some calculation. if we calculate the concentration of IgG of each colostrum with the volume that we need to give to these lamb and kids, if we have a poor colostrum of 20 grams per liter of IgG, and we are giving these animals 200 ml of colostrum, do you know what's the concentration that's going to be in a bottle of 200? 4 grams per liter. How we are going to achieve the target of 30 grams per liter of 30 grams of mass of IgG? We have to give 7 bottles. Is anyone going to go every time to the nursery to give 7 bottles to one goat? Anyone? No. What if I have a 30 grams per liter concentration in colostrum, and I'm giving a bottle of 200 ml? The amount of IgG present in this bottle is 6 grams per liter. To reach the 30 gram mass of IgG, again, I have to go to the nursery a lot, more frequent than I'm going. So that's why I'm aiming to these high bricks in the colostrum of the farms that I assist, for ovine and for goat, because I want a higher concentrated colostrum. I want to give the best concentration possible in one bottle of colostrum. Okay? So take a picture of this table, because you will not find that anywhere. Let me know when I can move on. Oh my God, I need to rush. So again, from the bovine world, how can I improve maternal colostrum quality? And I like to read whatever we have in any species, because I can go back and apply. So in this study, they had 2 maternal colostrum, one 30 grams per liter and one 60 grams per liter, and they add colostrum replacer to improve this 30 grams per liter maternal colostrum to a 60 grams per liter concentration. Sorry that I'm talking about numbers all the time, but this needs to be understood. We need to improve concentration of colostrum. And what they found on the serum IgG when improving the maternal colostrum with colostrum powder is that you can increase. So this is a calf with serum IgG of almost 12, and then it was improved to 20. Do you think this calf here is more able to fight disease than this one? Yes, right? So that's what we want with the small ruminant world. I did the same on farm, but I did enrich to a little more higher bricks in goat colostrum. And when I access the serum IgG of these animals, and I use the target of 15 grams per liter in IgG, in their serum, I found 93% efficacy on the transfer of passive immunity. So only 7% of my animals failed, which is a very good number. Very good. But of course, I aim super high because it's just me, you know. Next time I'll do 25 to learn how they go. So quantifying, when do you go on farm to collect blood of this animal to check if they had a good transfer of passive immunity? So according to studies, the best time to go is between 24 hours and 30 hours. In the bovine world, they did access this transfer of passive immunity, the peak, and when it decays. And what they found out is that the best time is between 17 hours to 72 hours. So if you are going on farm just once a week, make sure you are not collecting blood from not very old, but older animals, because you are like, it's useless. Take a picture, guys. Quantifying the transfer of passive immunity in goat kids and lambs. So I got everything that is available. If you know any paper that's available that's different from that, please come to me. and give it to me because I want to read. When you are assessing lambs, you can use the Brix refractometer, the total serum refractometer, and of course you can always do serum IgG by radio immunodiffusion, which is the gold standard for calves, goat, and lamb serum IgG assessment. So for the lamb and also the goat, using a Brix refractometer, I'm always aiming for the good, above 9%. And total serum protein, I have to confess that I don't use a lot. I use much more the Brix, because I use the Brix to assess the colostrum quality, and then I use the same refractometer to assess the animal. Okay? When to use a colostrum replacer? Basically, when you don't have maternal colostrum, you have a very high challenge. When you want to improve your maternal colostrum quality, you Brix it and it's 20. Are you going to just, okay, I'm going to use the 20% Brix, or you are like, oh no, I'm going to improve this colostrum. I always improve the colostrum. When you have poor immunological or microbiological quality, you can also use too, or if you have presence of disease, some disease that you want to eradicate from your farm, you can use a replacer. And what to use when you don't have any colostrum? You can use another colostrum from some dam of your herd, or you can have a whole bovine colostrum replacer, but has to be whole, because remember the energy requirements of these animals that I calculated to you. So don't give these animals a colostrum that doesn't have fat. These animals can acquire immunity, but they will lack on energy. I'm almost done. I put the pasteurization slide, but you can call thermal treatment as you wish. For the bovine world, we say that ideally you do 60 degrees Celsius for 60 minutes. We learned that the IgG concentration doesn't decay a lot, and the bacteria count decays a lot. So with the bacterial load lower, these animals can absorb more immunoglobulins, because the immunoglobulins are going to be available, they are going to be easily available for the absorption. For the small ruminant world, it's a bit different, because remember that the solids are much higher. When you pasteurize in a high temperature or for too long, they clot. So this is, I challenge you, if there is any research group here, please study more about that and give me solutions. I need them. Really. So the studies that I found, they used a lower temperature than in the bovine world, 56, 57, different amount of times, 60 minutes, 10, and 30. But look at this loss. Super high. Super, super high. So I have two take-home messages, 2 slides of take-home messages. First one, that colostrum is vital for newborns. They need to survive. We need to decrease this mortality rate. I don't want to, no, I'm not going to say that. But we really need to make something different. We need to change colostrum protocols. We need to help those animals. Colostrum is responsible for the passive immunity, so we need to give a very high colostrum, concentrated colostrum, that helps these animals to overcome infectious disease. Colostrum is the natural choice. It's very sad to go back on farm just to treat animals or just to calculate the mortality incidence, and it's super high. Where is Dr. João Costa? I think he's there. He's having a big problem in his facility. 30% mortality in the goat facility of the university. I hope that was not a problem to share. a good colostrum management will improve the growth of the animals, the productivity, and it will decrease newborn losses. And take-home message number 2, vaccinate the dam. That will improve the IgG in caprine and ovine. And I don't work for a vaccine company, guys. So really, take care of your animals. Test the quality. A Brix refractometer, it's a cheap, easy tool to use. Be quick. Give colostrum as soon as possible, the quickest you can. And give the right quantity. 15% of body weight is the minimum. Let's aim high. 20 is gonna be our next minimum. Give it clean, because it's super important. We know that bacteria doubles every 20 minutes. So if you collect colostrum and just leave it there in the bucket, you are giving a collection of bacteria for your animals. And quantify. Learn if your protocol is working. That's all that I had, but before, I need to acknowledge some people that helped me to build this talk to you. So this is Dr. Juliana Palhares Campolina. She was a colleague of mine in Universidade Federal de Minas Gerais, Federal University of Minas Gerais, Brazil. We worked together in the same lab, and she's amazing, so she's my co-author in this talk, so thank you very much, Joana. Thank you very much, muchas gracias pela organización del congreso, está muy, muy maravilloso. And also, I want to acknowledge some countries that are doing research in small ruminants: Gran Canaria, UK, New Zealand, Australia, China, Italy, Greece, Israel, Turkey, and the list goes on and on and on. But those countries, studying and publishing, they help us, our business grow, the community to grow, and we need to study a lot about small ruminants to understand. So thank you so much. Gracias, obrigada. Grazie mille. Dankjewel. Arigato, that's all I know. Thank you.
Thank you very much, Dr. Juliana Mer. Thank you. We will give you a small present. Thank you very much. Okay, we will continue with the next speaker.
Guys, if you have a question and you are shy, just find me on booth 23.
Yeah, okay, thank you. We are in time, so we'll continue with the next conference. The next speaker is Aurélie Montagnon. There are a chain in the speaker with the conference Nutritional Emulsifiers as a potential digestibility enhancer in the ruminant metabolism. Thank you, Dr. Montagnon. Go ahead.
Is it working? Yes. So, hello everyone. As mentioned, I'm Aurélie Montagnon. I'm also based in the Netherlands, working at Orpha, a company in the Netherlands, and I wanted to present you an abstract specifically on nutritional emulsifiers. First of all, I want to thank all my co-authors that you can see on the slide, and especially Professor Veerle Fieve, who is working at the University of Ghent in Belgium, that really designed the trial. Yes. So just to explain a bit, why did we do that type of study and why did we design it like that? We need to dive a little bit in the fat metabolism of the ruminant. I'm not going to dive really long because you are all very familiar with it. But basically in the rumen you have hydrolyzation of dietary fats by the rumen microbiota. And we also look at unsaturated fatty acids that are biohydrogenated into saturated fatty acids. But what interested us mainly was what is happening in the small intestine. So all the different free fatty acids or microbial lipids will be absorbed thanks to the combination of bile salt, but also pancreatic juice, for example. But we well know that the absorption overall in the small intestine can be variable depending on a lot of factors, the animal himself, the breeds, the health, the diet, etc. And in fact, if you look at different studies and literature, nutritional emulsifiers are typically used in monogastric to improve also this free fatty acid absorption in the intestine. So the idea was to, in fact, maybe nutritional emulsifiers could also support, for example, the bile salt to improve the free fatty acid absorption in the intestine of ruminants. But before testing that, we wanted to be sure that it will also pass the rumen. So here it was a goal to simulate the rumen conditions in vitro and to assess the impact of the emulsifier on the volatile fatty acids to make sure that everything goes well in the rumen. Our hypothesis in this trial was that the emulsifier will not impact either positively, either negatively, the ruminal VFA production. So this is the protocol. For all the different treatments, we worked in triplicates, and we first put in the flask a dairy cow ration. So composed, you see, on 30% grass silage, 30% maize silage, and 40% concentrates. So quite a typical diet, at least for the Netherlands. And then we added 5 milliliters of rumen fluid of adult fistulated sheep, specifically on the hay concentrate diet. Then we had 4 different treatments, so composed of different dosages of hydrophilic nutritional emulsifiers. And this emulsifier was composed of the active ingredient called glyceryl polyethylene glycol resinolate. And I just want to mention that specifically for the glyceryl polyethylene glycol resinolate, there was a specific chemical synthesis to make it extra hydrophilic. You see that we had 4 different dosages, so either non, 25, 250, or 2,500 microgram per milliliter. And just to mention, the 250 microgram was more the recommended physiological dosage in ruminants, and then we tested 10 times less and 10 times more. All these different dosages were then dissolved in dimethyl sulfoxide. And then in the flask, and to reach the 25 milliliter volumes, we just added the CO2 saturated bicarbonate and phosphate buffer. So after making all the mixing, let's say, we did an incubation at 39 degrees, so to recreate the rumen temperature, and this was done during 24 hours. We collected 1 milliliter, and this 1 milliliter was then centrifugated at 4 degrees Celsius during 15 minutes. The supernatant was filtrated, and then we did the analysis of the volatile fatty acids. As I mentioned, that was our goal, and this was done through gas chromatography. So with a nickel column and a flame ionization detector, so quite a classical protocol in that way. So you see here on the table the first result, first on the production of the volatile fatty acids. You see then in the treatment the 4 different dosage, and you can see specifically on the left, so the total volatile fatty acids, and there was no significant differences at any of the dosage. But if we look at the specific volatile fatty acids, you see a significant decrease of the production of butyrates only at the highest dosage of the emulsifier, so the 2,500 microgram. Looking at the other volatile fatty acids, it was not significant, but I just want to mention that numerically, because you will understand in the second slide, but that the acetate was numerically decreased and the propionate and valerate was numerically increased. Completely on the right, you can see also the apparent rumen degradability of the organic matter, and there was no significant differences for all the dosages. So this is for the molar proportion of the volatile fatty acids. And as I mentioned, when I looked at the numerical result, here you can see that it's significant for all the volatile fatty acids that I mentioned. So we had a significant decrease of butyrate and acetate, and a significant increase of the propionate and valerate only with the higher dosage of the emulsifier. So at the 10 times higher than the physiological dosage. So we wanted to, we brought in the different literature to look what could be explained in that and what could be the consequences. These are, of course, only hypotheses that I want to mention. But if we see the increase of propionate and decrease of acetate, it could be linked to methane. Of course, that's really a hypothesis that would need further studies, but I think that was worth mentioning. And we see that there is also a big relationship with butyrate and valerate linked to the metabolism in that way. So to look at the different results, in fact, the lipophilic emulsifiers, such as lecithin, that you might have heard already, are already quite proven both in dairy cattle and beef cattle, or also small ruminants. But we still have limited research in bovine, ovine, caprine, etc., with the glyceryl polyethylene glycol ricinoate that I just mentioned. So in this study, we didn't have any impact on the volatile fatty acid production or the pH, but there was an impact on the molar proportion at the highest dosage, so the 10 times more than the recommended physiological dosage. But if we look at the normal dosage, the 250 microgram per milliliter, we didn't see any impact on the volatile fatty acids. So it shows really a more neutral effect of this nutritional emulsifier, this hydrophilic nutritional emulsifier in the rumen in that part. And it would be interesting in a way to continue further those studies to see really the impact then now on the small intestine, as it could be interesting for the free fatty acid intestinal absorption. Thank you so much, and if you have any question, please feel free.
Thanks, Doctora. Any question for the speaker? No? Okay, it's interesting. Thank you very much. Okay. Dr. Marcello. Okay. Our next speaker is Dr. Marcello Guadagnini from Italy. The conference is Megasphaera elsdenii NCIMB 4112I capsule administration effect on growth performance in Holstein calves preliminary study. Results, sorry. Okay, go ahead, doctor.
Thanks. And thanks to the organizers for giving us the opportunity to share some field data about this novel tool that can be used in the field. And to my co-authors, some of them are here in the room, that did practically all the job on the farm. So first of all, let's talk about weaning, because sometimes we do a very good job during the lactating phase, but then when we approach weaning, something fails. And actually, weaning is just a big transformation for calves from pseudomonogastrics to fully developed ruminants, and it's a significant challenge. Some authors compare this transition to the transition that we see in a cross-parturition in dairy cows. So it's a significant challenge also because several organs need to switch their role, especially liver, and also the habits. We have several attitudes that need to be developed, such as the rumination, a lot of the salivation ability, and also we can see on the right side that we have a different role, size, and composition of the rumen. Actually, rumen and reticulum, they start to be very small at birth, and they really need to develop and be the most important of the four stomach. This rumen development is mainly driven by fiber consumption in size and muscle of the reticular rumen, and volatile fatty acids production and the fermentation inside the rumen drives size, length, and density of the rumen papillae that at the end will be the ones that will allow absorption of nutrients into the rumen. When this will happen is really different across different farms because of the management. So we know that as far as milk allowance is pretty high and calves are drinking milk, the intake of starter is pretty low. So in farms where they have a low milk allowance, they will start right away with a good starter intake because they need energy. In farms where milk allowance will be pretty high, there will be a great delay of starter intake so that this switch, when milk has to go down and concentrate has to go up, it's a little bit delayed compared to low milk allowance. But sooner or later, we will have these two lines that we see below that will be actually crossing each other. And we need a very consistent and high starter intake to have a healthy calves across this transition across weaning. Megasphaera elsdenii is not a new bacteria. It's a rumen native microbe isolating in sheep rumen in the UK by Sidney Elsden in 1953. It's a strict anaerobe, and actually it's one of the most important lactic acid utilizers. We have a lot of examples of lactic acid producing bacteria, but this works a little bit differently, consumed lactic acids, and will produce other volatile fatty acids. It's a strict anaerobe, which makes tremendously difficult to handle it, and we'll see that actually it's a big achievement. We now have the possibility to use it in a commercial way. Studies on cattle adapted to high concentrate diets showed high numbers of Megasphaera elsdenii. The point is, it's a bacteria that grows pretty slow, and to have a viable population, it takes about 3 weeks to establish a population that is actually able to manage rumen pH and to control the bumps in rumen pH. This is especially important in fully developed ruminants. What is mostly important in calves is the consumption of lactic acids and the production of butyrate. So it preferentially consumes lactic acids, but you will see it's not the only substrates, which is the primary influence of rumen pH. And it produces butyrate, but also propionic acids. We will focus more on butyrate because actually in calves, it will promote epithelial growth and rumen health. So this is very important. This bacteria was so interesting to microbiologists and researchers that actually since the beginning, since its discovery, it was attempted to use it for administration to cattle as a probiotic. But it was not easy. It's a strict anaerobe. Today, we will see the results and some data about a special strain of Megasphaera elsdenii called NCIMB 41125. It was isolated from a high-producing dairy cows in South Africa in the 90s, and it's a patented strain with a patented technology that allows to use it commercially thanks to the freeze-drying technology. Megasphaera elsdenii NCIMB 41125, it has a high specific growth rate, much higher than most of the natural occurring strains, a growth range of pH between 4.5 and 6.5, which is tremendously important because some of the strains, they don't hold on when pH is dropping so much. They are able to manage DNL lactate as preferential substrate. As you know, there are other lactic acid utilizing bacteria that are not able to metabolize both forms. It's capable of utilizing glucose and other carbohydrates, which is very important. Sometimes we don't have so much lactate to be metabolized in critical times. And it tolerates ionophores. Otherwise, it will be very difficult to use it in the field. So these skills make this strain very special and very interesting to be used on the field. There are already studies, published studies, where Megasphaera elsdenii, this NCIMB 40125 was used as direct-fed microbial or probiotics in calves. Previous studies showed that the administration of these bacteria led to higher plasma beta hydroxybutyrate, which is actually witnessing a higher butyrate production in the rumen, actually 7 days after its administration, which happened in this study at 14 days of age. Greater reticulum weight and papillae surface area, so bigger absorption, so you can see high butyrate production, greater development, and then which led to higher growth rate, high average daily gain, which at the end of the day is something we really want to see and what producers really want to see. As you can see, most of these studies were done with low and moderate milk allowance, and the administration was done at 14 days of age, because the starter intake was already high enough to guarantee a replication of the bacteria. So the study aim was to address the association between Megasphaera elsdenii NCIMB 40125 capsule administration, which is the form that is commercially available, among others, and growth performance during the pre-weaning phase in Holstein calves. The novelty of this research sits into the high milk allowance and a delayed Mega-E or Megasphaera elsdenii administration. So as I told you, previous studies were administering the product at 14 days of age. This will be further delayed due to the high milk allowance. So as you can see here, the test product was a small capsule, 3.55 centimeters long, that will actually protect the product from being exposed to the oxygen, that will dissolve into the rumen fluid, and containing 5 × 10 to the 9th CFU of Megasphaera elsdenii NCIMB 41125, commercially known as Lactipro FLX. As you can see, it's pretty easy to administer. It's very small and it stays into just one hand. It's very small for a calf. So this study was performed on a high-producing dairy farm in the Goiás state in Brazil, and 96 Holstein female calves born from January to May 2023 were enrolled into the study. They were randomly assigned to a treatment, Lactipro, with 49 calves, and a control group, Con, of 47. We had several weight measurements with a weight tape at birth, at day 30, which actually is the last measurement before the product administration. Then we had the product administration to the treatment group of 55 days plus minus 6, and the control group was untreated. And then we had a weaning measurement, which happened at 89 days plus minus 3. The nutritional management was the following. During the pre-weaning period, calves were housed in individual hutches and fed 8 liters per day, 2 meals per day of whole milk enriched with powder, with milk replacer powder to achieve 20% dry matter intake for the first 30 days of life. After that, the milk was gradually reduced to 6 and 4 liters until complete weaning. And during the whole period, the whole lactating period, the commercial starter feed with 23% crude protein was offered, and also grass hay as 5% of total dry matter were offered to calves. The initial weight was slightly higher for the control group, with 41.4 kilos on average, compared to the lact group, which averaged 35.9 kilos. And this difference was significant. But after 30 days of life, the weight didn't differ at a median value of 68 kilos. So actually they started right before administration, the weight did not differ. The treatment age actually, as it's a commercial study, and the farm was not huge, there was a considerable variation from 44 to 67 days, averaging 55 plus minus 6. So weaning age was happening, was averaging 90 days for the control group and 88.7 days for the Lactipro group, significantly different, but not a big difference, by the way. In the attempt to consider all these variables, we built a linear regression model accounting for, obviously, treatment group, body weight at day 30, age at weaning, and the interaction of treatment by weight at day 30 and treatment group by age at weaning. And what we saw is that the least square means for the ADG from day 30 to weaning were 115 grams higher for the lactopro group. And as you can see, it was with a very low p-value of 0.003. In an attempt to produce some variables that producers are more commonly or have greater habits to use, we also assessed the ADG from birth to weaning using the same type of model, and was 200 grams per day greater in the lactopro group. Similarly, we assessed the weaning weight, which was, again, with a linear regression model, yielding 8.1 kilos more for the lactopro group. So this study demonstrates that the administration of a capsule containing Megasphaera elsdenii NCIMB 41125 during the pre-weaning period is associated with higher growth during the pre-weaning period and higher weaning weight in Holstein calves. And actually is agreeing with other studies that even with different management, with lower milk allowance, they were actually yielding positive results on growth and on the rumen development. Thanks.
Thank you, Dr. Marcello. Question for the speaker? Yes, go ahead.
To me, the weaning age of the calves in your study at 90 days was a little long. They were on milk a little longer than what we usually have.
Yep.
Would you expect the treatment group to perform differently if they were weaned earlier? Would you expect to see less in the way of average daily gain and end weight differences?
Most of the studies that we have, it's an earlier weaning. This is a little bit late, and even if you see the nutritional management is typical of Brazil. Enriched milk, whole milk, enriched milk, it's something that is not common in all the countries. To be honest, we have tried with different conditions, even in other countries with earlier weaning, it yields the same results. The magnitude could be a little bit different, but actually, yes, it needs, wherever calves are approaching the weaning and they are leveraging more on the function of the rumen, we see at that time a lot of advantage to have more developed rumen. Because sometimes, especially in high milk allowance, we have the big issue, cows are growing fantastic when we approach weaning, and even after weaning, we have a post-weaning slump, and we have big issues. So this is a very nice tool to allow calves grow faster and be more consistent.
Okay, thank you. Another question? Yes, please. In the microphone, please.
Carlos Ambrosio Aguilar, del Centro Educativo Universitario Siglo XXI. Nosotros en la unidad pecuaria que tenemos, destetamos a los 3 meses. Quiero pensar que manejas que son 8 kilos más de peso al destete, que quizá no es menos tiempo, pero que sí genera una ganancia de peso. Mi pregunta es, el costo de ponerle esa cápsula comparado con los 8 kilos de beneficio, ¿en qué me ayuda a mí como ganadero? Porque nosotros generamos un alimento con un porcentaje del 30% de proteína que se le suministra durante la primera semana de nacimiento que empieza a lactar, se le agrega un suplemento que tiene el 35% de proteína, lo hacemos con soya y algunas sales minerales. Pero esto me permite desarrollarle el rumen, pero a mí me llama la atención, ¿cuánto cuesta esa cápsula? Y en el costo-beneficio de los 8 kilos que me da, ¿me genera una utilidad o un beneficio? Muchas gracias.
Could you please repeat in English, summary the question?
I understood it.
Yes.
Can I answer? So my answer is, well, if you want commercial information, you're more than welcome to join our booth at the lower floor, because every country has, I mean, the main goal of the talk is to illustrate scientific evidence on talking about the return on investment. To be honest, we saw earlier today some sober studies addressing how big is the benefit in milk production, like 800 kilos of milk across the first 3 lactations for every 100 grams. This was 200 grams more. So, well, it's hard to give straight calculation, but I think it's pretty easy to return investment with 200 grams more of average daily gain per day. So, and even if it would be less, and if it would be 100, it will yield a positive return investment. But more than happy to welcome you on our booth and have commercial information on the product.
Thank you. Thank you.
Okay, thank you. Well, thank you very much, Dr. Marcelo Guadagni. Our next speaker is Catherine Denholm from United Kingdom. Go ahead. Thank you. The conference is effect of feeding 5 days of transition meal compared with meal replacer on health status, growth rates, and dairy calves. Okay, thank you.
This is not the right slide, so I wait.
It's not that correct, right? Yeah, no, it's the same conference. Okay.
I can start while you find my presentation. I work at the university in Glasgow, and that's in Scotland. Someone in Mexico asked me where in the United States Scotland is. It's not. But we are doing some pretty interesting, well, we think interesting work on colostrum management in dairy calves. And this particular work that I'm going to present this afternoon is on transition milk feeding, which fits with some of the other stuff that's been presented on extended colostrum feeding. So probably best to explain that extended colostrum feeding can mean a feeding day 1 colostrum in an extended fashion, feeding colostrum supplemented milk replacer or whole milk in an extended fashion, or more practically for dairy farmers, feeding transition milk, which in this study we used milkings 2 and 3. So we were quite specific. You will have seen in the previous presentations that the concentration of immunoglobulins declines with time as we move away from the time that the cows calved. So we used specifically days 2 and 3. Sorry. Can somebody please help me with the slides? We could go straight to take-home messages if you want, or... Yeah, thank you. Okay, so Ryan presented some information on colostrum beyond immunoglobulin G, which is the concentration of immunoglobulin has been the thing that's interested people about colostrum for many years, but now we understand that there are many other components of colostrum, including oligosaccharides, which act as a substrate for beneficial bacteria and also prevent binding of pathogenic bacteria to the intestinal mucosa. Insulin and insulin-like growth factors, which work through the somatotropic axis to influence growth, cytokines and hormones, vitamins and minerals, maternal immune cells like neutrophils, macrophages, and leukocytes, and other immune modulating factors. And then I've included those other peptides, lactoferrin, lysozyme, lactoperoxidase, and trypsin inhibitors, which all are included in this, as Ryan described, perfect cocktail. And we know increasingly there are papers describing the effects of these, but it's an emerging area of science. And so we think that the way that the production benefits of extended colostrum feeding in dairy calves is modulated through some of this sort of perfect cocktail. Yeah, this is not working now either. Yeah. Okay, so the objectives of the study were to compare the average daily gains and health score outcomes between dairy calves fed an initial feed of 4 to 5 litres of first milk and colostrum, followed by 5 days of transition milk feeding, which I've already explained was milkings 2 and 3, usually pool mixed, and then the control group was calves just switched immediately to milk replacer, which is quite common on UK dairy farms. Next slide, please. Thank you. So we had 150 calves enrolled from 2 dairy farms in Scotland, and they received 4 to 5 litres of first milk and colostrum within the first 4 to 6 hours of birth, and then they were randomly allocated to one of 2 groups. The group 1 conventionally fed calves were immediately switched onto milk replacer twice daily until weaning, and then the group 2 calves were fed transition milk for 5 days, so 10 feeds. And we used the Wisconsin-Madison health score, which many of you will be familiar with, to score fecal score, respiratory score. You can leave it on that side, thank you. Respiratory score, and we, as part of the respiratory score, we scored cough, eye, ear, and nasal scores, and you can see a snippet of the Wisconsin-Madison scoring there. We also did take rectal temperatures, but this is in Fahrenheit, and we use Celsius in the UK. So all first feed colostrum and all transition milk was also sampled at the point of feeding and stored frozen at -20°C for subsequent testing. And calves were blood sampled for IgG. Yep, next slide, please. So in the lab, we used Petri films, which are the yellow grids that you see on this slide. We used these as a convenient method to measure bacterial contamination of the transition milk and the day 1 colostrum samples. They have been validated against more conventional plates. In North America, they've done that work. So they are really nice, easier to use because they don't require as many dilutions. Any of you who've ever tried to count bacteria in colostrum will know that you have to dilute it multiple times because a lot of times it's really just a bacterial soup. So this makes it slightly easier. And we measured the umbrella total bacteria counts, and then we also specifically measured coliform bacteria. And the reason we measured coliforms is because those particular species have been implicated in interfering with IgG absorption at the gut level. And then the middle cassette is a Triple J Farms radial immunodiffusion kit, which is a direct measure of immunoglobulin concentration in the calf serum, and we measured the zones of diffusion. We put 5 microliters of spun serum on the cassettes, and then we used the Brix refractometer, which many of you will be familiar with, to indirectly estimate immunoglobulin concentration in the first milk and colostrum and the transition milk. Next slide. Thank you. So these are the results so far. These are preliminary results, so we hope to publish this later this year. But you can see that the total bacteria counts and the coliform counts were very high in both the first milk and colostrum and the transition milk. And because this was an observational study, we were collecting data from the farms as it was done on farm. So the normal practices were followed, and it was, as I've already indicated, a real bacterial soup. And we fear that that may influence some of the production results which we hope to see by feeding transition milk in an extended fashion, because the calves were obviously being challenged by quite a lot of bacterial species, because again it was an observational study. But there was no difference in the serum IgG concentration between the calves in the conventionally fed group and the extended feeding group, because they were all managed equally at the start. They were all given the same volume at the same time of first milking colostrum. Next slide, please. So what we've seen so far with our preliminary results is that the calves fed transition milk for 5 days performed better than the conventionally fed calves in terms of mean fecal and nasal scores, but there was no difference in the cough, eye, and eye-ear scores between the two groups. There were also numerical differences in time to first health score for the fecal and total respiratory scores, but these were not statistically significant. And there were also no differences in the mean rectal temperatures of the calves. It did seem that the transition milk fed calves, grew slightly better than the ones that were conventionally fed. Next slide. So this colorful slide, you'll see the stars indicate small but statistically significant differences in mean fecal scores and mean nasal scores between the control and the extended colostrum or transition milk fed group, but there was no difference in other scores. Next slide. And the time to disease event, the dark bar is the transition milk fed group, and the lighter colored bar is the control group, and you can see that the time to event was slightly different. The next slide, please, shows the Kaplan-Meier survival curves, and the red line is the transition milk fed group, and the blue line is the conventionally fed group. So you can see that the time to event for the total respiratory score on the right side of the slide and the fecal score or diarrhea score on the left side of the slide are slightly different between the two groups, and the transition milk fed group performed slightly better than the conventionally fed group. So as I've already said, farmers are, and the Danish vet this morning explained that we're looking for practical ways to feed colostrum in an extended fashion. And the options for this include supplementing either whole milk or milk replacer with a powdered colostrum supplement or replacer, adding first milking colostrum to the whole milk, or feeding transition milk. The caveats to feeding transition milk include that you have to, as with first milking colostrum, be quite particular about the animals that you select as your donor cows for feeding. So, for example, Johne's disease cows should be excluded, or any animals that have undergone antimicrobial treatment. We don't advocate feeding waste milk. And the optimal time frames for extended transition or colostrum feeding haven't actually been confirmed yet in the literature. There's an indication that feeding a lower concentration of immunoglobulins and other colostrum components for a longer time is better than feeding high concentrations for a shorter time, but we don't know the exact parameters of that yet, so that's an option for further research. And industry recommendations, both in the UK and internationally, should move towards extended colostrum or transition milk feeding for dairy calves in the first days of life, rather than abruptly switching them from the first feed of colostrum onto milk replacer, since there's a growing body of evidence, including this work, extolling the health benefits of this, including reducing calf hood disease events, particularly diarrheic disease events and respiratory disease events, and also average daily gains and mortality. Next slide, please. So, yeah, I'd like to thank MSD Animal Health for sponsoring this work, the Clyde Vet Group vets and their technicians in Scotland for collecting the samples and visiting the calves every day to health score them, and particularly Nicola Gibb and Aurélie Montagnon, who are sick of my WhatsApp messages. Then the other staff at the University of Glasgow, Neil Evans and Michelle Bellingham, Anna Montero and Becky Orr, who are my technicians in the lab, who are also endlessly patient. Thank you, and I'll take any questions.
Thank you, Dr. Catherine DeHaan. Any question for Dr. Catherine? No? No question.
Okay.
Okay, no, thank you very much. Okay, let me check. Thank you. Is Dr. Gregory Chambers in the room? All right. Okay, thank you. Sí. 15 minutes. We are waiting 5 minutes to continue.
The sky calls to us. What will become of humanity?
We are waiting 5 minutes to continue. And when finish this part of the program, the lunchtime, we are invited for everyone to the lunch in the ground floor. So we are wait 5 minutes, only 5 minutes, please.
we are wanderers, and we are one.
No tienen videos, ¿verdad?
Determinación de la calidad del calostro. Calostrómetro y refractómetro Brix. El objetivo de este video es mostrar el procedimiento detallado de la evaluación.
Continuing with the program, now the next speaker is Dr. Gregory Chambers for New Zealand. The conference is the concentration of copper in the life over time with different methods and doses of injectable copper supplementation. Okay, thank you very much, Dr. Chambers. Go ahead.
There's a term you might have heard of called combinatorial explosion. It describes when you get a large number of options or combinations as the size of a problem grows, and it's used in computer science and psychology, genetics. And I'll give you a trivial example of it. This is something that happens to me if I go to somewhere like Subway sandwiches and I have to decide what I want to get. First of all, I want a sandwich, and then they give me the option of 3 different types of bread, and then is it fresh or toasted? And then what kind of cheese do you like? And there are 4 types of cheese, and so on, and so on, and so on. And that list expands so that it's so big to get you to the sun. Now, for most of you who are normal, that's probably fine, but for someone like me with a small brain, that's quite stressful. And that's a little bit similar to what is behind the study that I'm going to present this morning. In New Zealand, and I'm sure for most of you, there are a lot of options for trace element supplementation, including copper. And it can be quite hard as a veterinarian or an animal health advisor to work out what to do. Still some bugs, but it's all right. We've got multiple products. We're quite lucky in New Zealand, and probably the same for you. Lots of different options, but they have different routes of administration, different actives, different formulations. And then in New Zealand, where we're mostly pasture-based, we generally can't give supplements through the feed in a total mixed ration, so we might give it through the water, or by injection, or by other means, by drench. But complicating that is if you send your cows away off the dairy farm for the dry period and you rely on water administration, then you need to give some other form, and maybe they need to have an injection before they go away so that they don't become deficient over the dry period. Now, as a veterinarian, the farmer asks you, what should I do? Should I give them an injection? How much? Which one? And it can be quite hard to decide. And as a vet, of course, you're concerned about safety. What dose should you give? How long is it going to last? Will it carry them over until they re-enter the herd and can consume that water? So that's the basis to this study. And I'll just be honest, I'm a little bit of an imposter here. I did the analysis of the data. I didn't design and run the actual on-farm part of the study, so I'll do my best to explain it. But our friend in the South Island, John Spearpoint, wanted to find out what actually happens to the liver copper concentration of cows given various supplementations. So what we did is we wanted to enroll 80 cows, 20 in each treatment group, and we wanted cows from farms that had normal, adequate liver copper concentration, because we didn't want the study confounded by deficiency or toxicity. And we define normal as having a liver copper concentration of 150 to 1500 micromoles per kilo. So he went out and just randomly picked some cows on 3 dairy farms and ended up enrolling 2 dairy farms with 40 cows on each farm, and we randomly allocated within farm. We also took some bloods before enrolment too to look at plasma copper. And we split the cows into 4 treatment groups. One group was a negative control group, and the other 3 groups had an injectable copper supplement. Groups 1 and 2 had the same product, CopperGuard, calcium copper EDTA, but at 2 different doses, 2 mils or 4 mils. And group 3 was a different product again, multimin, containing disodium copper EDTA, but also zinc, manganese, and selenium. And as I said, the 4th group, control, they had no treatment. And what we did was do a bunch of liver biopsies, because we wanted to look at the dynamics of liver copper concentration over time. But before they had their treatments, at day -7, all cows had a baseline liver biopsy taken, and then after treatment at days 3, 14, 21, 42, 56 and 70, all of the cows had a liver biopsy. So we had repeated biopsies in these cows, 6 of them. From an analysis point of view, the objective was to look at the change in liver copper concentration at each point compared to baseline, because that's normally distributed, and just used regression and accounted for the repeated measures with a random effects model. So what did we find? Well, first of all, the treatment groups were balanced in terms of age. We actually blocked on age, so they should have been balanced. Plasma copper was balanced, and so was the prior liver copper concentration. We had 2 farms in the study. We wanted to enrol more than one farm so that we could see the effect of farm on our results. And they were balanced for age and plasma copper, no surprise there. But they did have different prior liver copper concentrations. Farm 1 had a significantly higher concentration on average in the liver. Moving to the change in copper concentration compared to baseline. On the left, that's just the distribution of change at all time points for all groups, almost normally distributed, but you can see some cows actually dropped. And then on the right is the unadjusted change across all time points for each treatment group. And what you can see there is the purple group, which is the negative control group, they were less likely to have an increase and more likely to have a decrease, therefore, than the other 3 groups on the left. But you can see that all groups had some cows that had a decline in liver copper compared to baseline. And numerically, it looks like the 3 treatment groups were higher, particularly group 2, which had the higher dose of calcium copper EDTA. But that's just a preliminary look. Then we adjusted for the other factors, prior liver copper concentration, age, etc., and found that the higher the cow's liver copper concentration was prior to the start of the study, the less her liver copper concentration increased with supplementation. So we're seeing that there's potentially diminishing returns with supplementation when they come in with higher starting points. Also found an interaction between treatment and time, as you might expect, because the effect of treatments will vary over time, but also with farm. So we had a 3-way interaction going on between treatment, farm, and time. So rather than show you a big long regression output, I'll just show you the predicted liver concentrations at each time point. And I have to do this by farm because we've got an interaction with farm. So at the top, with farm 1, they compared to farm 2, farm 1 had more of an increase over time, including the control group. They had an increase in liver copper over time, and there were numerical differences between the treatment groups and the control group. They were all numerically higher, but those differences weren't significant. Now, farm 1 is the farm that started with the higher liver copper concentration. So that effect of prior liver copper concentration existed independent of the farm effect. On farm 2, down the bottom, you can see that they didn't increase as much and they started at a lower point, but the higher dose calcium copper EDTA group had a significant elevation compared to the other groups. So at day 3 already, their liver copper concentration was higher than the control group, and then at day 14 it was higher than all 3 other groups. At day 28, higher than the control and the lower dose calcium copper EDTA group, and at day 4, higher than the control. So we saw a significant increase out to 6 weeks after supplementation, just for that one group. So summarising that, going back to the combinatorial explosion, we weren't necessarily trying to fix that altogether, and I'm not sure that we have, but there are a few things that we've learned from the study. First of all, that 200 milligram dose, the 4 mil dose of CopperGuard, did significantly elevate liver copper for 42 days compared to the negative control group and at other time points to the other groups. But that was only on one farm. On the other farm, the differences were all numerically there, but either just due to study power or the effect size, it wasn't significant. We also learned that confirmed that the higher the cow's prior liver copper concentration, the less it will increase through supplementation. So that suggests that perhaps, as well as those cows benefiting less from supplementation, you also get diminishing returns because it's harder to drive an increase. But the main point is to tailor your advice to the farm. And so as a veterinarian, one of the things to think about here is you can't give a generic recommendation for all of the farms under your care. It needs to be farm specific, and you need to take some liver biopsies from 10, 15 cows and find out what's happening, work out what they're doing in terms of supplementation through the season and what options they have over the dry period. So that's really the main point. It's got to be farm specific. That's it. I'd just like to acknowledge a few people. John Spearpoint, who did the work on farm in the South Island. Matt Wells, who's here from Verbeck, who's supported the study and supports a lot of work in New Zealand. Thanks, Matt, and Verbeck as well for the sponsorship. My colleague Emma, and of course the farmers and the cows who participated in the study. So I'll wrap up there and welcome any questions.
Okay, thank you. Thank you, Dr. Gregory Chambers. Any question for the speaker? Yes, go ahead.
Hey Greg, how you doing? Just a question specifically around the copper. Were there any differences between the two farms on soil type and any interfering factors that may influence the absorption of copper through the diet?
Yeah, good question. That's one thing that we didn't do in the study is look at pasture and soil information. So we don't know what other factors there might have been with things competing for copper. We know that they were both supplementing in the water, and Farm 2 fed the cows palm kernel extract, which is high in copper, and they had been doing that prior to the study and during the study. They didn't change anything. But yeah, those other things in the soil and in the pasture, we don't know.
Thank you. Another question? No? Okay. Thank you. Thank you, Doctor. Sí. Okay. Sí. Te la mando. Okay, we have the last conference with this part before to the launch. The next speaker is Dr. Manuel González Ronquillo. There are a change in the program of the speaker. The conference is ruminal fermentation and ruminal ammonia concentration in beef cattle supplemented with urea and urea-protected additive. Okay, go ahead, Dr. Ronquillo.
Thank you very much, Mr. Chairman, and thank you for the committee for the invitation. Well, I would like to present the next presentation, which is it's ruminal fermentation and microbial and ruminal ammonia concentration in beef cattle supplemented with urea and urea-protected additive. As you noted, normally the ruminal bacterials convert the nitrogen non-protein sources into microbial protein depending on the amount of nitrogen supplemented and the amount of energy available for the microorganisms. In this case, there are many nitrogen, non-protein nitrogen sources that partially can replace another kind of expensive true protein like a soybean meal or canola meal, depending on the kind of diet that we would like to feed the animals. And in this case, we try to use feed-grade urea, which is the most used in the market for supplement nitrogen source for the microorganisms. And in another phase, there are another slow urea releasers that can be protected for slow this urea more slowly into the rumen. The basis is that if we feed the animals with some other sources of slow urea release, the risk of alkalosis can diminish in some cases depending on how we feed the animals. In this case, for example, for this study, we decided to feed the animals in one pulsed doses at the beginning with a higher amount of slow urea releasers and feed-grade urea, and see if the risk of alkalosis could be diminished, or in this case could be give a better animal performance in the animals. Then the objective of the study was to conduct an in vivo study to determine the effect of feeding feed-grade urea and slow urea release as a replacement for true protein supplements like soybean urea, which is a many conventional diet. And also we use another control diet. What it was that we didn't supplement any other nitrogen source as a control diet. And in this case, we decided to see or determine the nitrogen ammonia concentration pattern among the time and see the animal performance also. Then we use 5 beef cows and we distribute we distribute the animals in a Latin square design, in a completed Latin square design. And the animals were performing for different diets. Diets contain 60% of corn silage and 40% of concentrate diet as a fresh matter. And the diet was control diet, soybean meal diet, feed-grade urea, in this case was urea normal. The other trends were optigen, burette, and high urate, which are used as a slow urea release sources. The diets in general, with the exception of the control diet, which contains lower protein, the rest was isoproteic and isoenergetic diets. Then the animals were offered a total mixing ratio once a day at the 8 a.m. and the experimental period lasted 30 days. Then the first 23 days was for adaptation to the diet. And after that, we decided to make the data collection for intake, refusals, feces, urine, to see the digestive performance. And the last 2 days of each trial, we take the animals with the ruminal cannulas and we take ruminal liquid into the rumen at the serum. We'll see it later, but in order to determine the nitrogen ammonia liberation and pH. The amounts of feed and refuses were measured daily to determine dry matter intake, but as I told you, I would take only the data for 5 days for determining the digestibility trial. And the last 2 days, we take urine and refusals for collecting the 10% of the samples with frozen. Later, we analyze the samples for the digestibility trial. For the ruminal ammonia concentration, we determine before feeding, and after that at half hour, an hour, 2, 4, 6, 8, 12, and 24 hours before feeding the animals in every period. And also the pH was determined by duplicate. Samples were determined for nitrogen ammonia, for pH. And also, for example, for the feed tubes and the refusals were determined for the chemical composition for dry matter, organic matter, crude protein, the nitrogen fiber, acid deuterium fiber, and lignin samples. Basically, as I told you, it's a 5x6 incomplete Latin square design. And finally, for statistical analysis, we make by the covariate the initial live weight for corrections for the live weight. And we determine the dry matter intake, grain feed ratio, average daily gain, variables for the analysis. And in general, we found, for example, that there were not difference for the initial and final live weight in the animals. Also, we didn't find difference for the average daily gain and the gain feed ratio in the whole treatments. For the intake and digestibility, animals with urate and high urate eat a little bit more compared with the rest of the treatments, and also for soybean meal. And in the digestibility coefficients, the diets for burette and optigen and soybean meal were higher compared with high urate diet. When we notice, for example, the nitrogen intake and excretion and nitrogen balance, and we express it in grams per kilograms of metabolic light weight, we didn't found difference for nitrogen intake and nitrogen excretion. Only for urinary excretion was a little bit lower for high urate compared with the rest of the treatments. And the nitrogen balance was lower for high urate compared with urate diet as a slower urea release. When we made the in vitro gas production for see the fermentation pattern for the different diets that we analyzed, control diet has always the higher gas production, and the lower gas production was for feed-grade urea diet, followed by soybean meal. When we try to show the ruminal fermentation ammonia dynamics among the time, the interesting in this table is that always high urate depress the ammonia concentration in the whole time compared with the rest of the diets. If you notice, for example, feed-grade urea always had higher levels in general, and followed with, for example, for Optigen or Butyrate, that at the end of the way, we didn't found that they depress or inhibit the nitrogen ammonia liberation into the rumen compared with high urate, for example, or even with the controlled diet. This is the patterns, and for example, when we notice, for example, with Optigen or also with Butyrate, it has a higher concentrations ammonia at the first hours after the administration of the doses. And in these points is where we can find the risk of alkalosis into the animals in any feeding. When we notice, for example, with the controlled diet, or also, for example, for soybean meal diet, it doesn't give higher peaks, for example, which is similar, for example, with high urate that depress the nitrogen ammonia concentration among the time. When we correlate this data for the pH value, it's similar, for example, always it has doesn't affect the pH into the rumen. Normally, when we found an alkalosis risk, the pH of the liquid ruminal is above the 8 value. For example, in this case, we have ranges between or near to the 7. It's very stable. And we can notice the pattern of the pH among the diets. A little bit lower for some diet times with the controlled diet compared to the soybean meal diet, but it's not so high, the difference for the pH. As a conclusion, we can say that in this study, replacing soybean meal with feed-grade urea or slow urea release did not alter the final body weight, average daily gain, feed conversion rate in beef cattle. Then any diet effects were also found in the nutrient intakes, while Optigen and Butyrate was superior in terms of digestibility of dry matter and nitrogen balance. Then the lack of difference between feed-grade urea and slow urea release in animal performance must be see if it worked to use it or not. In this study, we didn't see any difference at the use of this kind of slow urea release. Thank you very much.
Thanks, Dr. Ronquillo. Any question for the speaker? No? Okay, all is clear.
Thank you very much.
Thank you very much, Dr. Ronquillo. We have the lunch time at 13:55, in a few minutes. Okay, we invite you to the Cow Nutrition and Rearing Symposium, sponsored by SCCL in the afternoon, and we have now the lunch in the ground floor. Okay, see you.
It has become harder to take your eyes off a screen, to enjoy life as it's happening. Lost in the digital realm, we've become slaves to the screen's seductive glow. As the baseline drops, we rise above the screens, above it all. Because in the end, we are all just pixels dancing and pulsating. society is dead.